Good morning, and welcome to the NIDDK Central Repository 20th Anniversary Workshop, promoting secondary research to accelerate medical breakthroughs and innovation. It's 10 o'clock, so we'll start the meeting. Next slide, please. We have a few general housekeeping reminders for the meeting today. Participants will be in listening mode and will not be able to ask questions verbally or use the chat function. Please submit your questions using the Zoom webinar Q&A box. Messages will be received in real time, and the session moderator will select questions to read to the speaker during the Q&A portion of each presentation. For technical support, for help, please use the Zoom webinar Q&A function, and someone will reach out to you shortly. And finally, both days of the event are being recorded, and the recording will be available in about 1 month on the NIDDK website. And now, Sharon Lawler.
Thank you, Mark. Hello, my name is Sharon Lawler, and I will be serving as MC again today. So welcome back to the NIDDK Central Repository 20th Anniversary Workshop, Day 2. I'm also joined by 2 repository colleagues who will be helping with the Q&A and the chat throughout, Ms. Dee Cooper and Ms. Elisa Fujimoto. So please keep us busy with the questions coming in for our different panelists. And with that, this morning, I'll turn it over to Dr. Rebecca Rodriguez, the Program Director for the repository, for a few opening remarks to kick off this second day of the workshop. Rebecca?
Good morning. Thank you, Sharon. Thank you, everyone, for joining us. I would like to welcome all of you to the second day of the NIDDK Central Repository 20th Anniversary Workshop titled Promoting Secondary Research to Accelerate Medical Breakthroughs and Innovation. We thank all of you for your interest in learning more about the repository resources, and thank you all the speakers and panelists who graciously accepted to participate in our workshop. I want to remind you that this workshop is being recorded, and the recordings and the slide decks will be made available on our repository website. Today we have a packed agenda, as you see here. We will start the day with a moderated panel discussion. We will follow that with a keynote plenary session, and then we'll move on to our second workshop for this series. And then we'll, in the afternoon, have a lightning talk from some of our data requesters, and end the day with a panel discussion from some of our funded researchers and other experts that will talk about expanding the NIDDK data ecosystem. And with that, Sharon, I pass it over to you.
Thanks, Rebecca. So as Rebecca said, we're going to start off the session with a panel discussion titled Fairization of NIH-Generated Resources, Meeting the Intent of the 2023 Data Management and Sharing Policy. And as moderator for this session, I have the pleasure of introducing Dr. Eric Schultes. Dr. Schultes is an evolutionary biologist with a data-intensive research focus and has held previous academic appointments at the University of California, Los Angeles, the Whitehead Institute of Biomedical Research at the Massachusetts Institute of Technology, and the Santa Fe Institute. Since 2018, Eric has been the FAIR implementation lead at the Go FAIR Foundation and is now senior researcher at the Leiden Academic Center for Drug Research. Eric is co-author on the original publication of the FAIR Guiding Principles and has, since 2016, been working with a diverse international community of stakeholders to develop FAIR data and services. This work included the development of the first accredited FAIR awareness training programs for executives and practical introductions to FAIR data stewardship for frontline data producers. Eric is also co-developer of numerous FAIR maturity evaluation services and is the architect of the now widely adopted 3-point FAIRification framework that is deployed in numerous international settings. So Eric, welcome, and thank you for taking time to moderate this session.
Great. Thank you very much, Sharon. Appreciate the kind introduction there. And also, thank you, Rebecca, for the invitation to chair today. It's always an honor, I think, to have an ability or an opportunity to work with an NIH panel like this. So very happy to be here. I would like to lead off. Well, first, I think the second slide here in my own slide deck is, so if we advance the slide, so we have the session panel. So first thing I would like to do is introduce our panel members today. First, just a kind of a high level view, Robin and Marianne, I think, are representing the stakeholders who have an interest in standards and preparing standards that can be widely used, whereas Jen and Todd are coming from really the data producer and data curator levels. So there's slightly different stakeholders represented on the panel here today. But let me tell you a little bit more about Robin. She is the product owner of the NIH Common Data Element Repository, which in GoFair anyways, we're a big fan of. As a member of the NIH CDE Task Force and the NIH CDE Governance Committee, she advocates for the use of CDEs, these common data elements, in the NIH-funded biomedical research applications. And she also supports NIH users who are interested in developing and sharing and reusing these CDEs. She holds a Master's of Library Science and Information Science from San Jose State University. Marianne Henderson is the Senior Advisor for Division Resources and Biobanking in the Division of Cancer Epidemiology and Genetics at the U.S. National Cancer Institute and the NIH. She supports a large program and contract management and infrastructure planning for molecular epidemiology. Marianne is a past president of the International Society for Biological and Environmental Repositories, but she continues to contribute in the science policy and education and training with the organization. She has received a ISBER Funders Award and the ISBER Distinguished Leadership and Service Award for this continuing dedication to global biobanking. Then we have Todd Valerius. He is on the research faculty at the Division of Renal Medicine at Brigham and Women's Hospital at Harvard Medical School. He is also a co-investigator for the Atlas D2K program, and that stands for the Analysis, Technology, Leadership, Administration, and Science Data to Knowledge program. And there's a repository platform there that brings together contributions from different NIDDK-funded consortia with cross-study data linkages and analytic tools. So I think Todd in his practice is very much a practicing person with FAIR principles. And then lastly, we have Jennifer Dara. She is a senior research data management consultant in the Center for Data Visualization Sciences in the Duke University Libraries. In this role, she provides training and consulting services designed around the support and coordination in research data life cycles. She also serves as one of the service coordinators and curators for the Duke Research Data Repository and as a member of the Data Curation Network. She holds a Bachelor of Arts in Psychology from Westminster College in Pennsylvania, and also a Master of Library and Information Sciences degree from the University of Pittsburgh. And so that is our panel for today. I want to say just a few introductory remarks regarding the FAIR principles themselves. So if we could have maybe the next slide. Yeah, so that's a little bit about me, GoFair Foundation, and the LACDR. Next slide, please. Yeah, so there is this, so I just want to get a little bit of background on the session today. So there's this NIH and in fact, the NIDDK's strategic goals, and I think that's informing the structure of the session today, and I think what you'll see for the rest of the day today. There's obviously this interest to maximize the value and impact of NIH-funded resources. There's an interest to advance the biomedical and data ecosystems from technical level. There's an interest to enhance the data resources that will facilitate research. And of course, that's all for catalyzing discovery and improving health outcomes. And it's, of course, the data sharing policy and the FAIR principles, I think, have a direct impact on these strategic objectives. In the next slide, I wanted to highlight two, kind of the two cardinal directions for the work today. There's the NIH data management and sharing policy, so I just want to highlight that. And I think the theme that will resurface over and over today is this idea on secondary research. And certainly at the NIDDK, there's a very strong interest, and I'm showing a link to the web pages here on resources for research, and the idea that the resources at the NIDDK are meant to be reused and repurposed. But then that raises a question: what does it mean for data sharing or for the application of the FAIR principles for secondary reuse of data? And in the next slide, I think we can see that the stated goal then for today is really to discuss these practical examples of the application of FAIR principles to biospecimens and other data assets that are for secondary uses and that are intended to meet these NIH DMS policy guidelines. So that's the, I think the goal for today. Just a few remarks on the FAIR principles themselves. And so in the next slide, I just wanted to highlight that, from my day job at the Go FAIR Foundation, we always start from kind of the basics here. These are the FAIR guiding principles. They're 15 one-liners. And when they were originally published, there wasn't much really said about how they should be interpreted or even implemented. So in Go FAIR, we've been spending a lot of our time trying to help communities articulate that for themselves. From a very high level, we can see that metadata is really important in FAIR. I'm highlighting that in yellow. But we also see there are some aspects of FAIR that are really focused on technical aspects. So we might look to professionals in IT and computer science, and these are the red principles I'm talking about. So these are infrastructure-based components that we look to IT professionals. That's in contrast to the blue principles, which are, I will call, collective decisions, or these are the domain relevant standards that have to be agreed upon within domains. So we need both to power FAIR. We need kind of the generic infrastructure components, the machines, but we also need the practicing scientists to help us develop the language and the interest in the data. In the next slide, I also wanted to just highlight that in the original publication, FAIR was not just an aspiration for humans. Of course, it's wonderful if humans can do FAINR, but the reasoning behind the principles was that we wanted to enhance the ability of machines to automatically find and reuse data. So FAIR as an acronym is really for the machines. So in the next slide, I try to say, I summarize FAIR by what we're trying to do here is find and implement principles whereby we can automate the F and the A and the I and the R so that the machine can do more of that work for us. And then finally, in the last slide, just another hint that in the Go FAIR Foundation, we've been able to make a commitment to the FAIR principles pretty rigorous. And it's really just sticking very closely to the FAIR principles themselves. We often work with groups to develop what are called FAIR implementation profiles, which is simply going through each of the FAIR principles and asking, how is it that you actually implement? What are the technical components that you use to implement F1, F2, F3, et cetera? I would say that the closer we get to actually implementing FAIR, the easier it becomes to actually answer these questions. And I hope that's a kind of a good background and context for what comes next. So I would like then to ask each of our participants here, our panelists, to give us a small overview of their perspective on the data sharing policy and the FAIR guiding principles. We're going to start with Robin. And Robin, we'll give you the floor for 3 minutes like everyone else. And we would like very much to hear your view on these things.
Okay, thank you, Eric. Thanks for the introduction. My intro slide here, excuse me, my title slide mentions the NIH CDE repository, but I'm actually going to start with a more general introduction to common data elements. Next slide, please. So this definition of common data element is from a 2021 request for information that NLM issued, the National Library of Medicine, about the use of CDEs in NIH research. And I think it's held up pretty well with a lot of use over the past couple of years. And by this definition, there are 3 key parts to a common data element. The first 2 are the question and the response. And you could say that those 2 together, they make the data element. And the third part of this is systematic use across different sites, studies, and trials, and that's what makes it common. Of course, that puts the C in the CDE. So just to rephrase this another way, in other words, if research is being conducted across different sites and studies, and the same information is being requested in the same way, same question or same variable across the different sites, and the responses have to be in the same format or come from the same answer list, then those sites and studies are using CDEs, and the data collected across those sites is interoperable. So just a note on what I've seen in practice with some different institutions, some funding institutions, and I'll just pause and say at NIH, I've seen there's a wide variety of sort of maturity levels with CDEs. Like some institutes are much further along than others. It's fine. Everybody understands the importance, I think, and everybody's working on it. So what I described, you may or may not recognize from your own work, but I've seen funding institutions that require researchers to use certain core CDEs for collecting data that's likely to be very common across many studies, like demographics or medical history, that kind of thing. Then researchers are free to add their own unique data elements on top of that, on top of the CDEs for their own research questions. And then sometimes that funding institution might also provide some supplementary CDEs that are not required, but if a researcher is going to collect data about, say, alcohol use, the institute recommends that the researcher collect that data using a certain CDE or a set of CDEs. So if you have multiple studies and they all ask, how many drinks containing alcohol do you have on a typical day when you're drinking? And then the response can be, from an answer list with choices 1 to 2, 3 to 4, 5 to 6, and so on. If all the studies ask it that way and have the same answer list, those are CDEs. And if some studies ask it that way, but some studies maybe they ask the same question, but the response can be any number. And then some other studies ask, how many drinks containing alcohol do you have per week? Those are not CDEs. Okay, next slide, please. So hopefully from that brief example there, you can see that when researchers use CDEs, their data is going to be more interoperable. It allows apples to apples comparison across different sites and studies. It also makes that data more reusable because it allows aggregation and reanalysis of data from those studies. And of course, the CDEs themselves can be reused. That's what makes them common. That's what we want to happen. And this, over time, this makes the data more reproducible as well. So one quick final note, it's best when CDEs are linked to other data standards, such as terminologies and codes. They make the meaning and the usage of the CDE more clear, and they result in even more interoperable data. Data interoperability, reusability, reproducibility, these all facilitate, as you probably already know, collaborative research and open science. And I think I may be close to out of time, so I'm not going to talk about the NIH CDE repository right now, but please feel free to reach out to me afterwards if you have any questions. We have some free training resources available, live training coming up in October if you want to know more about CDEs and how to use them in your research. Thank you.
Great, thank you, Robin. And yeah, nice to know there's an entire repository out there waiting to be explored. Thank you. Marianne, would you then like to give us a brief overview from your point of view?
I think my video should be showing, but it's not showing on my screen. Oh, here we go. Okay, I'm Marianne Henderson. I work at the Cancer Institute in the Division of Cancer Epidemiology and Genetics, and I am going to represent a biobanker's point of view on this panel discussion. So next slide. So our division has a mission to discover the causes of cancer and inform the means of prevention. We do this through transdisciplinary epidemiologic and genetic research. These are long-term investments and cross the different areas in epidemiology that might affect someone that may be on a pathway or trying to prevent a pathway to cancer. Next slide. So in our division, we're very aware and working toward fair practices in epidemiology. In fact, we just published this article in the American Journal of Epidemiology that you can see here. Data sharing is essential for reproducibility in epidemiologic research, replication of findings, pooled analyses, in consortial efforts especially, and to maximize the study value. So that's where we are really focusing. We know that there are barriers to fair practices in terms of confidentiality, cost, and incentives. They often limit the extent of sharing and the speed of data sharing in epidemiology. If we follow these fair practices, we make our data resources findable with the necessary metadata, using CDEs, accessible to authorized users, interoperable with other data, and optimized to reuse this. We are building many different tools within our organization, and I think we'll be talking about that on the panel. And but this paper is out there for anyone to see where in the epidemiology community we're moving. If you go to the next slide. So within repository science and biobanking, because we're a molecular epidemiology shop, we have to think about our biobanking activities, the materials we're collecting, and the associated data, and the procedures for use of that activity. And we are all focused on the conformity of those practices within our repository activities. If you click once, our repository is about 15 million specimens at the NCI at Frederick. And so we need to make sure that these samples not only are usable for our primary aims, but also for our secondary aims. And if you click again, we're focused on quality. So we're looking at SOPs and reusing SOPs as appropriate. We're looking at conformity of those practices to those SOPs. We need reproducibility, especially in rare disease. Many of the cancers we have to collect from many different sites and many different research groups to be able to study our specific rare disease so that we can get comparability. In order to do this, there are two different large standards activities within the repository world: the ISO standard for biotechnology, biobanking, and the general requirements for biobanking. There is the main standard 20387, as well as some auxiliary standards. And these are available internationally, and NIH participants across the government helped form these. These were informed by the ISBER best practices that is actually about to release its 5th edition, and that will probably come out next month. We were part of that, and we were really focused on the metadata and the standards that need to fall into best practices of repositories, not only in the US, but in our partner organizations around the world in high income and low income.
Marianne, I'll just interrupt and let you know that we have to finish.
And I'm finished.
Perfect. Perfect.
Thank you.
Super. Thank you very much, Marianne. We're going to ask Jen to present her point of view next.
Okay, hi everybody. So just to give you a little bit of background of the kind of role that I have, and essentially why, you know, so many libraries these days have people who help support research data management, is because research data management is caring for, facilitating access to, preserving, and adding value to research data throughout its life cycle. It is not something that just happens at the very end, which I think was something that was happening there for a while. Data curation is the process of ensuring that data sets meet the FAIR guiding principles. One of the things that I want to make sure, because I'm a little bit different than some of the other folks on this panel, is that, you know, I have particular expertise in social and behavioral sciences data, so I can do a huge depth of curation there. But for Duke, I'm a generalist, and I have to be able to support all disciplines. Next slide, please. Thank you. So as part of the Data Curation Network, Duke is a member organization, we're a group that share curation staff to allow for deeper and more specialized curation than you would have locally. So for example, if somebody had something that was human participants data, socio-behavioral research, I might be tapped to do that because I have a depth of knowledge in the data formats and the documentation, the types of things necessary there. One of the things that we do share, though, is a model for how the data curation process should take place. And this is called the curated steps. Because we found that, you know, in our organizations, mostly academic, a lot of researchers understand how to meet the FA in FAIR, but that the I and R can be a bit more challenging. So how our curated workflow goes is we have to check files to make sure that they open. Sometimes people will give us things without file extensions. That can be interesting. We want to review the submission package to ensure that it's understandable to somebody of equivalent knowledge, you know, and that it's complete. And if perhaps it could even extend beyond to more generalized knowledge. We want to request any additional information for submission optimization and address any accidental errors in the submission. We want to assess that metadata to make sure that, you know, at least for those of us with generalist repositories, that at least it adheres to minimum viable standards, or when it's more of a disciplinary type repository, you know, that people know which ones they should be doing, that it adheres to that. We transform files from proprietary formats when necessary to open so that it's more interoperable across different platforms. And then we also make sure that we evaluate the final data submission package and the repository record to make sure that it meets all of the requirements of FAIR. And finally, we document provenance and curation processes so that we can see any steps that were taken to change the data. Let's see. So one of the things that's challenging for the researchers in the Data Curation Network is because a lot of us are academic institutions, we do have some specialized ones, is that we see all kinds of people and they don't always necessarily have a dedicated disciplinary data repository to use. They might not also have a specific metadata standard in their field to follow, or they just are completely unfamiliar with what metadata standards actually are. So one of the things that we often push very hard on is having contextual documentation that makes it much more easier for an end user to understand what data is in front of them. Next slide. Thanks. We were asked to provide, you know, some examples, which was hard for me to think about at first. And I thought about some common questions and one particular issue that I encounter. One of them being that researchers really are unfamiliar with some of these terms. It's interesting because in the library world, we've tried to avoid being too jargony for our patrons, and tongue in cheek here, our funding agencies have picked up our jargon and have given it back to our patrons who are like, I don't understand what metadata means. What is a repository? Can't I just put this up on my website? Isn't that good enough? They don't necessarily understand persistent identifiers and some of those things. So there's a lot of education that goes on. How much data am I supposed to share? We have people who generate petabytes of data per project, and they're like, well, I can't necessarily share all of this. What am I supposed to do? And the biggest thing for me, again, is reminding folks that data sets, when you're putting it in a repository, needs to be able to stand alone as a published object. You can't always rely on research articles for context because they don't necessarily give you variables or values or any of those kinds of things in the article itself. So you want to make sure that that data set is well documented so that it can stand alone, not only for reproducibility purposes, but also to further additional research. So that's my part. I think I kept us on time.
Well enough. Thank you very much, Jen. That's great. And last but not least, I think we can hear from Todd now.
Yeah, so I just want to make a couple of comments without slides, but I just want to say that Jen hit on exactly the experience that many bench researchers, I'll say, hit upon and run into when we get started. And that is, where to start and how to start. It's heartening to hear that there are, from like as Robin mentioned, providing some of the CDE elements. I think that's more common, at least in the medical center setting, in clinical studies, and less common in some of us basic scientists and the type of work we do. So when we were starting the goodmap.org project, we certainly had to generate metadata that we felt was appropriate. And our guiding principle has always been, if you wanted to use somebody else's data, what would you need for metadata? That's kind of a loose way of doing it, and it's a little too loose, to be honest with you. So as metadata standards start coming along, we start to incorporate those. And then about 6 years ago, when Carl Kesselman at USC came on board the project as a data scientist coming from other fields, like even planetary research, the idea of FAIR was introduced to us, and we certainly adopted it quite readily. That said, even though this is our guiding principle at Atlas D2K, GoodMap and RBK consortiums, and in the KPMP consortium, it is still difficult at times to decide what we really need to do, and we've had to pick it apart. I think for us, our main thing is working through the vocabularies and trying to figure out how best to capture these things, especially when it's a brand new technology. When we started we were working on the goodmap.org project, all of a sudden single cell sequencing was there. And as a new technology, there wasn't a lot of guidance for the first several years on what we should be collecting and how we should be sharing it. So you have to sort of be very nimble in this regard. That said, imaging's been around a long time. Nonetheless, it can be very challenging because there tends to be many standards available to use, and collapsing those down into things like OME-TIFF and things like that many of you are probably familiar with is an extra layer of work for the data generating labs. Nonetheless, it's important to make that more generally accessible. I remember when I was a postdoc, which was a while ago, but the early 2000s at Harvard, we did have the librarians come to us and talk to us about metadata and images and tried to engage us in sort of working through this. And it was a foreign concept at that time. So I think it's very, very important that the academic librarians and librarian systems are more involved. And it's nice to hear that is happening. I also would like to say that one of the clear values to me of having the central repository biospecimens in general is that, I'm very fortunate at Brigham, we have a very large biobank. But many researchers throughout the country will not have access to that type of biospecimen repository. So it's very important that we support and continue to argue for these types of things to be available, and making sure that new researchers understand what's available. And I think the FAIR process is a way to do that and connect data. So those are the thoughts I wanted to share from a bench scientist. I think that having the policies on data management that NIH has put forth is really encouraging, well, let's say forcing in a good way, institutions to make sure they incorporate this type of thinking into the day-to-day activities, which is fantastic. So thank you.
Great, thank you, Todd. And that concludes the initial thoughts from our panel. So it's leaving us some time now for Q&A. I see there's already a question in the Q&A, but I encourage everyone who's listening, if you have a question, now would be a time to log your question in the tool. But I will lead off with maybe some kind of warm-up questions here. So to the panel, would you like to comment on any particular tooling or, I don't know, methods, training programs, et cetera, that you might be developing that would help researchers to more rigorously adhere to the data sharing rules in the NIH, or to say the FAIR guiding principles more generally? So just an open question if anyone would like to step in and take a stab at that.
I will briefly, sorry. One thing that we've tried to do at Atlas D2K is, as our data generators are collecting their information for publication, we've encouraged them to create what we call collections. So if the data is in our repository, this is data, not biospecimens, but data. We provide a facility so that they can collect those, whether they're images or single cell, RNA-seq, whatever, into essentially a little web page that links out to all the different pieces of data. And we generate DOIs, the digital object identifiers, to include in their papers. Investigators that have taken advantage of this find that they get a lot of activity and attention from their papers through to the data. So that's promoting the data out there. You can read a paper, see the data, and go straight to the data, the exact data. I like to call it the longest amount of supplementary data you can possibly have, because you can link to all the additional specimens. We find this is a very powerful way to get the data out and in use, which I think is a great way to do it, to encourage investigators. Now, that requires preparation when you're working through and struggling to get that manuscript submitted. But if we can convince people to do that, I think it's a great way to engage readers of the manuscript.
And you find that the academic publishers are often okay with this?
Very happy with that. Very happy with that.
Very nice.
I can add to that. In our EPI activities, we have always had a repository of all of our publications out online. We're in the final stages of developing what we call the PDR. It's a publication data repository that allows curated access depending on the research question, the authorized use. And that will help support providing access to the data sets that are within our publications. Those are linked to the study itself, and the study is described on our website so that you understand really the basis of the study, because our biospecimens are only as valuable as the data associated with it. So we want to make sure that that's clear. We don't want to just put a catalog about, oh, we have this many prostate cancer specimens. That's not the value of the specimens that we keep in our repository.
Well, and that's related to another question that we were thinking about before we launched the session today, and that's and I think Todd has already alluded to this as well. So there's the classical idea of a data set, maybe tabular data that's in a spreadsheet, but there's also images and annotations on images, and there are biospecimens of all kinds of varieties. What does it look like then to create data sharing pathways or standards, and let's say FAIR principles or FAIR guidelines for this much wider class of research objects, in addition to, say, just Excel spreadsheets? Because there's also software, there's workflows, there's all kinds of components that are valuable from the research output. How do we handle, and in fact, is it possible to handle in the same way these other kinds of research objects?
May I jump in?
Yeah, please.
So I just wanted to mention, since we were somewhat talking about tools, is again, the data curation network, again, we're a network of folks. We deal with all kinds of data in all kinds of formats. One of the things that we've done is we have had a series of workshops where we're teaching other information professionals how to curate. And right now there's going to be a pilot workshop. My colleague has a grant to do more specialized curation with different kinds of data and software and things like that. But one of the things that we've developed, and I'm putting it in the chat, let me see if I can put it for everybody, are a set of primers, which goes in depth into different kinds of data types and formats for people to consider how to meet the FAIR guiding principles when they're doing curation of those objects. These were designed more for information professionals, but we have found that they can be just as beneficial for researcher use as well.
Great, and they cover this variety and diversity of research outputs, I guess.
Can I add on to that? Around the world and at NIH, there are catalogs of specimens. Some of them are quite well defined. Some of them are you go to another place to get more information. But those are growing in use around the world. In repository world, we know that biohoarding is a big issue, and what it does is it also reduces the sustainability of repositories, and we're finding that that they're probably the percentage of use of specimens at high is about 10% of specimens in repositories around the world. So it's a real problem. I don't know if you're aware, but the National Science Foundation actually has, it's not only a data sharing standard, but they also are including a specimen sharing standard within some of their granting activities.
Super. Very nice. Robin, I have a question for you about the CDEs. Because I think the way you presented this, it all makes eminently good logical sense to focus on the problem this way. My question would be, if you see and can identify, any barriers to kind of institutional uptake, or, what are the things that might slow down the reuse of CDEs?
That's a great question. Everybody, I think most people, if asked, would agree in principle that CDEs are a great way to approach research, and you can't deny the benefits of them. In practice, it's as with any data standard. I think you find like everybody agrees that standards are great and we should all use them, but nobody wants to change what they're doing. So when you have groups that have been established and doing things a certain way for a long time, it can be hard to move them closer together to coalesce around a standard. Sometimes you ask for obstacles. I'll say one thing that does help with that is sometimes there are sort of standards coming down from an even larger organization, or something like, for us, the OMB standards about race and ethnicity, for instance. Like those can sometimes be like a sort of higher level directive that everybody has to sort of follow. That's one. There's also a perceived, I think sometimes there's a perceived burden that this is going to be, that if you have to ask, if there are CDEs that have to be incorporated into a study, it's going to just raise the burden on the participants. You're going to be asking them more questions, but I don't think that necessarily has to be the case. I think it's just, these are hopefully like questions that you would be asking anyway, but you'll be asking them in a more standardized way across the projects.
Yeah, so there may be, I don't know, strategies you have in mind. We're sort of running out of time now, but I'm sure you must have strategies in mind for overcoming some of these, or kind of working around some of these constraints and helping organizations adopt CDEs.
Yeah, well, we do. We do these sort of talks where we talk about the high level, like the benefits of them, and then the real world applications. The way that other groups have been using them quite successfully for some time without putting any undue burden on participants or on the researchers. They can actually save time when you're trying to set up a study. if you're using something sort of off the shelf, that's already created. And it certainly speeds up the work for the data coordination centers downstream when they're trying to harmonize things.
Absolutely. So there's lots of nice advantages to promote.
Yeah.
Very good. Thank you very much,
Can I
panel.
add
Yeah.
to that?
So I will look also to our MC to see exactly how much time we have left.
I believe that you were right at time, Eric.
Okay, so Marianne, I think we're probably going to call it a day at that point. And I would hand it over back to Sharon then.
Yeah, so I want to thank you, Eric, and all the panel members for actually having this great session and discussing, bringing these different perspectives to talk about how we move towards fairization of our resources. So thank you all. And with that, we're going to move on to the next session, which is featuring Dr. Yasser Tarabici, and he's going to actually talk to us about secondary research, breakthroughs in innovation, an informaticist perspective. And so it's my pleasure to introduce Dr. Tarabici. I believe he is on with us. Dr. Tarabici, do you want to turn your video on?
It's telling me that my host has stopped it.
I believe that your host may have stopped it. So can the host turn it back on? There you are.
There I'm up. I'm up. Thanks.
All right. Right.
You guys hear me okay? Just want to make sure I'm coming across loud and clear.
I can hear you loud and clear. So
Excellent.
let me just take a minute to introduce you to the audience, and then I will turn it over to you. So Dr. Tarabici is actually the Chief of Medical Information Officer at Ovation. He's the Director of Clinical Research Informatics at Metro Health, and also an Associate Professor of Medicine at Case Western Reserve University. Dr. Tarabici is a board-certified pulmonologist, intensivist, and a clinical informaticist with over 8 years of experience in leading and implementing health information technology solutions that advance research, care delivery, and clinical outcomes. As the Chief Medical Information Officer of Ovation, he guides the development of an innovative and an integrated platform that enables seamless collaboration across multiple electronic health record systems and healthcare organizations in the virtual first space. He also serves as the Director of Clinical Research Informatics at the Metro Health System, where he launches and supports data-driven tools and initiatives for research and operational gain. As an Associate Professor of Medicine at Case Western Reserve University School of Medicine, he mentors and teaches the next generation of clinical informaticists and data scientists. And with that, Dr. Tarabici, I'll turn it over to you, and I'm really looking forward to your presentation.
Thank you so much for that introduction, and thank you for having me. I'm a little late to the game here. Will I be asking for next slide, or can I share my own slide so I can control? Is that an option?
You can take control, I believe. Mark, do you want to confirm that he can just take control?
Yes, if you wanted to advance your own slides, yes, you can just start to share your screen, and then it'll come up on that.
I'll do that.
Okay.
I think the whole next slide thing gets a little tiresome at some point, right? This should work. How's that?
There you go. Yes.
Okay, great.
Perfect.
Wonderful. So again, thank you for having me. I'll start with my disclosures. So I do have some research funding and consulting fees from Beckman Coulter, particularly around the development of sepsis early warning systems, so unlikely to be related to this talk. As far as things I'm hoping to cover with the group today, I've been given 30 minutes to kind of think about innovation and breakthroughs in the secondary data source space, and particularly how it relates to health data. And so what I'd like to do is present some ingredients for secondary data analysis success, think about some upcoming and ongoing innovation opportunities and breakthroughs in that space, and particularly around the ability to unlock data, progress or advance analytics, and then talking a little bit about some of the platforms that are being evolved. And at the end, hopefully point out a couple of obvious ethical and privacy concerns that arise as we talk about this space. So I know that you guys have been talking about data and secondary data for a while now. We don't need to beat this up too much, but the sources are plenty and the volume is growing quickly. At least based on this projection that I have on the right-hand side, by 2025, we think that the compound annual growth rate of data for healthcare will get to 36%. That's compound annual growth, and it's already larger than most other industries and faster in terms of growth in manufacturing, financial services, media and entertainment. The caveat, of course, being that it is extremely siloed across the board, and so getting it all together and harmonize is one of our biggest challenges. When I think about ingredients for success in the secondary health data analytics space, I start thinking about the data at its source, that it is of high quality, very well curated from a metadata and contextual perspective, that it is standardized. And I caught the discussion at the end of the last talk, so I think that that's something you guys have obviously been thinking a lot about. Then I think about the next step, which is bringing it in together with other data sources, other organizations, for instance. And that's where we start talking about concepts like semantic interoperability and how we can deduplicate patients that may be present in more than one database or registry. And obviously the big one is de-identification to make sure that we're not sharing protected health information. And then putting that all together is the presentation layer that sits on top of this. And that's really the platforms that aggregate and present the data and or provide really nice statistical overlays and methods and pipelines that users can extend. So it's all about getting high quality data together to the experts in the space to gain insight and generate new findings based on data that we already have. And just to it's worth talking about semantic interoperability. The notion there is it's exchanging data with unambiguous shared meaning across systems. And so you can think about that like a lab test where hemoglobin A1C of 7 carries over to another system or database. And it's clear that that is a hemoglobin A1C of 7 units accompanying and context present in terms of when this happened, outpatient, for instance, and what time it happened. Make sure it's obviously connected to the right patient and that it's mapped correctly otherwise. So pausing there, now taking kind of a step back as a clinical informaticist in the space, I think about where we've been and where we're going, and namely thinking about opportunities and risks that have developed along with technological advancements in the space. And I see a couple of opportunities. The general buckets I think about are standards and interoperability and how those can help with the first two steps of this process. Obviously, artificial intelligence and machine learning have been around for a little while, but with more recent strides and acceptability, they've been percolating into this space. And they've been really touching all phases of this process. And then at the end, I'll talk a little bit about platforms. I've had been lucky, I should say, to have experienced a number of these platforms through either different vendors or otherwise. So I have a bit of a sense of the lay of the land for where we're at there. So I'll go back to my comfort zone here. A lot of the data obviously comes out of the electronic health record. And really, this has been the case for a while now, but we do consider, unfortunately, that electronic health records are data traps. The High-Tech Act of 2009 was incredible in that, well, we all use electronic health records now. Unfortunately, during that development, we ended up extremely siloed, and we ended up with varying local standards at each individual organizational level. There's a lot of data model and quality variability, even within users of the same proprietary system. So if you have the same EHR as your neighbor across the street, your data looks completely different. As a result of that, and also because a lot of smart people have had the foresight, we've developed really good standards and data models and data messaging, and you'll recognize some of these: Loink, SNOMED, HL7's, FHIR. The problem is organizations, specifically healthcare organizations, have spent a lot of money on the development of these systems. And so it's a lot of effort and ask them to switch their data sets or data models over to another standard, work on interoperability. And even when they do switch to another standard, it turns out that interoperability is not necessarily assured. And so give you a little bit of perspective about some of the efforts. A couple of research-based data marts and collaborations that I'm sure most folks are familiar with, OMOP and PCORNet are the two that I like to focus on. So OMOP is the Observational Medical Outcomes Partnership. It's part of the Odyssey Open Science Collaboration. They've got the data model and a number of repositories and networks that they've set up. An example is, for instance, just to keep the international perspective going, the European Health Data and Evidence Network, or EDEN. They tend to have rather epic names. PCORNet is based on the FDA Sentinel common data model, and it's got a couple of networks, research networks, where organizations have converted their data to OMOP, and that they share the data either at large or for a specific initiative. And I've given examples like Insight in New York City, or the Greater Plains Collaborative, which includes 8 states. So the really awesome thing about this is that these groups have been crowdsourcing and developing standards. They've been developing and evolving ETL tools, or extract, transfer, and load tools, and thinking about how to harmonize all of these different sources together. And they're really great, exciting, and excited communities. So it's always awesome to see what they're doing, and it's always awesome to be part of their meetings. The issue really is that the effort contained in doing this, it's considerable. It's a lot of time and money, and unfortunately not a great return on investment for organizations that are more focused on the operational demands. And the other unfortunate piece of this is that quality and fair principles are not always guaranteed, and we don't even necessarily have a consensus or benchmarking on what that actually would look like. On the right hand side, I've just shown you a figure of the rise in the number of publications attributed to the Odyssey collaboration over time. And you can see that it's rising, but I would love to see more than, say, 100 publications a year for such a large organization. To give you a sense for where quality and fairness and fair principles, which I heard you guys talking about as I walked in, not being guaranteed even based on a common data model. I've just showed you a sampling of the literature. And on the left-hand side, you have 2 plots, the one on the left showing vital sign outliers in a 13-member PCORNet network. And you can see that really for vital signs, there are not a lot of illogical values, maybe up to 2.5% for some sites, some better than others. And those are the A through M letters at the bottom. On the lab, from the lab perspective, I would say that there's probably a lot more bad matches. And you can see a high level of variability where the LOIN codes, which are your lab identifiers, are not matched in an accurate fashion. On the right-hand side, I've included some data from EDEN, or an OMOPified database in Europe that I mentioned earlier. And this is basically a comparison of the FAIR's FAIR framework for FAIR, showing that really they failed at a scoring system of up to 25. Both 2 Dutch ICUs contributing data to EDEN did not meet high-level standards for fairness. I've included this plot here to show you the process where the data goes from beginning to end. And I'm just going to shout out here, because I'm sure there's some Odyssey folks on the call. The tooling provided by this community is amazing, and it does a lot to help these organizations and these analysts get their data into the common data model. But there's still a fair amount of human input that goes into it. Usually an analyst in a moment thinking about whether or not they should map something to one observational framework or another, or one LOIN code to another. And that's essentially where a lot of the discrepancies come in when we think about what happens even after you've standardized to a common data model. Moving beyond what's happening in kind of the crowdsourced and common data model sourcing perspective, we obviously have, there's obviously a role, I should say, for regulation in the space. And so we promote standardization and operability through regulation in the US. A lot of you may have heard of, or been exposed to, at least in practice, the CMS's meaningful use, which evolved into promoting interoperability and has since been sunset. What's arisen out of that is the encouragement of health information exchange and a lot of digitalization, like e-prescribing, for instance, which again suggests that you are mapping data to a common standard that is understandable, by another entity. Even then, so even in things like the consolidated document architecture standards, it's clear that the mapping's not great and interoperability is far from achieved. Moving on from there, we have the 21st Century Cures Act, where we've got prohibition of information blocking, which again is release of information, is not interoperability. Although there's been a bunch of development in leaning on FHIR resources as a messaging standard for interoperability between healthcare sets and systems. And those of you in the space may be familiar with TEFCA, which is the Trusted Exchange Framework and Common Agreement that's arisen from the 21st Century Cures Act. And its idea is really, it's a voluntary initiative today to provide or generate a national health information exchange network, and several vendors have already signed on to do that. So some more promise in this space. I'll tell you a little bit about some of our own experience here. So we've been working, we have Epic as an electronic health record vendor, and we've been working with them for a couple of years now. And the idea here is EHR vendors are really best suited to solving the mess that they essentially or effectively started. And so they are experts in the systems that they created. They understand the variability in the data models that are out there based on the development of their system over time. And we think, at least as a healthcare system, that it's really on them to fix this problem. And so we pushed back on Epic to develop this interoperability network based on current health information protocols, health information exchange protocols. And so the idea here is, as a healthcare institution, we're already exchanging data with other healthcare institutions for patient continuity. And this is a lot of development supported by the regulatory and compliance space, and basically pushed into action by users of this system. We're using that network to interchange or exchange data, consolidate a single patient journey, and then present that data to researchers on what's now called the Cosmos platform. And what I would say about Cosmos, without spending a ton of time talking about the architecture, is we were able to accumulate more than 200 million unique patients in 2 years. And I think it's obvious why this scaled so quickly. You're using pre-existing agreements, a health exchange network, and data standards with a relatively robust patient deduplication mechanism provided by the vendor to consolidate all this data. It's essentially an easy on button for users of the same platform. And we learned a lot in the process from the Odyssey and Percornet communities in terms of how do you take care of common data models, how do you harmonize, how do you present systems that create semi-autonomous mapping, and how do you manage quality control. So obviously those things, and on top of that, a level of community engagement provided by users of this electronic health record. And so our conclusion for this paper that we presented was Cosmos success was really just due to its low barrier of entry. It was really that easy. A couple of examples to show that the data is good. For that same publication, we talked about how you can use Cosmos to basically look at asthma prevalence by weight across the US. And at this time, there were about 100 million patients on the database of the platform. And we compared that to data from CDC and Haynes, and we showed that we can show these differences in gender and asthma and race, gender, asthma, and obesity across the board and over time into more recent data. And that's the other amazing thing about this data set, is it's really up to date as to up to about a week from the time of a query. And so this is chronic disease surveillance. We talked about syndromic surveillance and how we can kind of use it to sample flu testing across the nation. And obviously you have the COVID spike there in April of 2020. We've even taken this further to look at differences in COVID spikes by states and showed how that correlated to initiatives at the time. And, obviously all this follows on the tail of COVID, where there was a massive push to develop interoperability and more health information exchange. And I think Cosmos just happened to sit there at the right time. We've done some health services research examples, like prescriptions for antibiotics for upper respiratory tract infections. Again, comparing NAMSIS data sets to data you can extract from Cosmos to show that overall antibiotic prescribing has gone down in kids and adults over time. And again, extended the data to recency to say this is still consistent as a signal. And then HPV vaccination compliance by race. I'm not going to spend too much time here, but the idea is you can look at HPV vaccination across demographics and look at first dose versus follow-up dose, and that's consistent and extends data that's already present. And then MMRV and febrile seizures, similar idea. We can use vaccine safety data link and review, lots of data Obviously, that's a good data source, but we think that Cosmos and that kind of a collaboration complements that, and we saw similar findings here. So I think one of the strong points here for Cosmos is that it's got this Care Everywhere ID, which is a probabilistic patient matching algorithm to identify a patient that does not rely on a centralized master patient index. And I've included some studies here to show how such a concept really is rather robust, that for the vast majority of time, the connections are made. And that's a really big limitation that a lot of platforms where the queries come to your data that they struggle with, right? It's that de-identification, and I'll talk about that in a little bit, or the deduplication, I'm sorry. I'll talk about that in a little bit. Some of the limitations I've seen with Cosmos and our experience with it, it really only pulls in structured data, but there's talk of expanding that. It only provides cohort counting. So how many patients have A, B, or C followed by D over some period of time. It tends to rely on numbers and some basic statistics like, well, what is the median age of that group? So the tool's kind of limited at the outset. There's also this notion of preventing re-identification using a statistical model. That is, as you add present variables, which can identify patients, the more unique the variables like address or area deprivation, right? Area-based measures that would hint at someone's census block group, for instance, in the United States, the more that you're likely to re-identify somebody. And so what they end up having to do, or we ended up having to talk about, is capping that to some degree to say, we have to limit the amount of demographics you can pull out so that you can't re-identify not just patients, but also sites that are contributing. And so that ends up really limiting the amount of analytics you can do. The complexity of the platform's evolving. They've just released a line level database, which again limits a lot of the data to prevent re-identification. Still haven't seen a whole lot from there. And the kind of the biggest caveat really is that the access is just severely limited, and that's essentially by design. So if you're a public health entity and you'd like to understand data in Cosmos, you have to work with somebody who's got access to Cosmos, and that's the caveat that was built into the contracting to give the healthcare systems a little bit of relief in signing up, really, that they're going to hold back the data as much as possible. Now, I don't know that everybody feels that that's appropriate, but that's where it started. And that initiative obviously that can evolve, and the initiative can evolve. So zoom out a little bit here and think about some of the constructs we've been talking about some of these kind of aggregation services and how they've evolved over time. On the left-hand side is a schematic for Cosmos. The idea there being really, you all have to have the same electronic health record. You have to flick a switch where the vendor can just ping the electronic health record directly and then siphon up the data and deal with the harmonization and data quality on their end with some feedback from the user. So we call that essentially, great deduplication, yes, super low effort, but absolutely no control. You flick a switch and it's all or none. And then obviously your sample size is your sample, I should say, is highly biased because you're relying on institutions that, for instance, in this case, pay a lot of money for their electronic health record, and that's not going to be the case for at least half the country. In the middle ground, I would consider an appliance-based mechanism where they set up a machine or a service behind your firewall, ping your data resource, try to work with you on the mappings to standardize and harmonize the data, and then essentially send off data based on their proprietary data model back to their central site. This is an example of this is Trinetics. They've been rather successful in the space, I should say. They do have deduplication algorithms based on a similar mechanism as the Epic folks do. For this, the moderate's a little bit more effort. The effort is a little bit more moderate. And the control, same idea, it's kind of all or none. You're either contributing or you're not. But the benefit here to the Cosmos model is that you can add differential EHRs. The final piece is a little bit more what the crowdsourced community has had to come up with, which is each organization, regardless of what their health source, data source looks like, has to map their data to a common data model with all the errors, that come out as a result of that. And by the way, they can do this. They can hire a second or third party to do this. So it's not always the organization doing this, but there's still some cost involved in the effort. And then they can potentially preferentially release data to these databases if and when they are okay with the query or the model or the framework that they've signed up to contribute to. So it can be kind of an on, or it could just be we're only responding to certain queries or contributing data to very specific circumstances or data sets. So you can see that that's a high effort, high level of control situation. And you can see why that's an organization that's difficult to achieve for every site, especially when most of these healthcare systems are really just trying to get by. So moving along a little bit of the spectrum from standards and interoperability, I want to talk about some of these more recent developments in artificial intelligence and machine language in the aggregation and processing step, and well, really actually the data cleanup as well. I don't want to beat this up too much. I think it's fairly obvious that one of the biggest innovations in the space is that AI and ML can unlock unstructured data. And it's probably nobody's surprise that at least in healthcare systems, the majority of the data is unstructured and unfortunately worse, uncaptured, which is, an informaticist cries every time healthcare data gets deleted. But data on machines like telemetry, for instance, these beeping monitors that go off in the background, that's often auto-pruned and flushed, gone, never to come back again. And actually more often than not, even in healthcare, as a general principle, we've been minimizing the amount of excess data we collect, which obviously hurts these efforts, but really more expense for patients if you want to put them on telemetry if they don't actually technically need it. Not going to get too much into vision. So pathology, radiology, dermatology, really robust AI development there. Plenty of literature and even some, I would say market approved and regulatorily approved mechanisms for using AI in that space. If anyone's thinking about going into pathology, radiology, dermatology, I would reconsider unless you want to drive the machine that does your work. Audio and video, I'll skip to those. I'm not going to touch those. I'll focus more on text. The richness of language that we have in the electronic health record, for instance, and how natural language processing and more recently large language models come in to play in this space. So I think it's a fairly obvious reason why or how AI can support data harmonization. As I'd mentioned already, most work today is manual. So you have an analyst or someone who has a little bit of an inkling of what they're doing, both on the data technical side and also the healthcare side, and they are mapping concepts to an end game of a common data model. Obviously, there's not going to be a whole lot of consistency. So AI can provide that consistency, good or bad, right? It can make the same mistake over and over again, and that's a problem. Adaptability to new data types and data creep. So these common data models are not set it and forget it. They actually take a lot of maintenance and monitoring from a quality control perspective and a remapping perspective. Scalability, obviously it's hard to do this at each individual site. If you could do most of it at least using an artificial intelligence, that would be phenomenal. And I think with the explosion of some of these transformer-based large language models, it's been really exciting to see what they can do like out of the box, zero shot, meaning no pre-training or nudging. They can basically do certain things in this space, and I'll get to a couple of examples in an interesting way. And all that kind of boils down to cost reduction. So, and scaling it, less human being involvement, unfortunately, cutting out the middleman, if you will, which is the classic tech play, I think can provide more in terms of harmonizing data both within organizations and between organizations for greater interoperability and obviously more powerful analytics. And to give you a couple of examples for what the large language models have been doing in the space. Some of this work, admittedly, that I'm presenting is in preprint, at least the figures, and I'll mention those when I get there. But if you think about the data that goes into an electronic health record, there's narrative data, like this is a patient that has, this many problems, recently had, for the example I give on the left-hand side there with the figure, an amputation, left above the knee amputation because they're diabetic. Large language models can parse through that, extract these key principles. And you can see that where they say the patient is Mr. Ham, 57, has these diagnoses and has an observation of an A1C of 12 based on a narrative, and then create an HL7 message, which is a standard, at least standard today, interfacing message for health information technology systems, where I can feed this, what was a narrative, into objective pieces of data and observations, like this is a lab, this is a diagnosis, and it does it correctly. Again, this is not a model that's been trained or tuned to do that. So that's literally out of the box. Another example is some of that mismapping we talk about happens all the time in routine care when I'm interfacing data with another healthcare system and they tell me they have a hemoglobin A1C of 500 milligrams per deciliter. Well, obviously there's a problem there. Now, as a human, I can tell that that's probably the glucose. Obviously, at scale, there's a lot of potential mismatches that can occur. And the example that I've given you on the right-hand side, the bottom right-hand side, is really presenting the HL7 message to a large language model and asking it, is this mapping correct? What is it probably? And also beyond that, actually, what is the loin code that I should map to this? And there's the answer. It's no, glucose, and 2345-7. And again, these are out of the box, no training. So imagine what they can do with a little bit more pre-training, fine-tuning, and some better prompting. The figure that I have on the top right just gives you a sense for what the recent literature has done with large language models. And the biggest thing, obviously, is NER, what we call named entity recognition. And that's where, frankly, they're the most powerful. It's exactly like the first example, which is what is the DICD? What is the, what are the objective findings that I can pull out of this note? Does this patient have heart failure, for instance? It can go on to do more things, like, for instance, predicting the risk of a disease, but I'd be really nervous about that one at this point. I don't think we're there yet. Another really cool large language model play, data de-identification. And so that figure on the top left gives you a sense for kind of where we've been in taking this unstructured data in the electronic health record, text typically identifying identifiers and then removing them. We start with manual, we move on to rule-based. Is this a name? Is this a date? And then more recently, current standard is natural languaging-based processes, and that the standard here, the industry standard, is more than 99% accuracy. Well, it's really interesting, and this is again another preprint here where the authors have shown how you can do this in a large language model, on ChatGPT specifically. Zero shot, out of the box, no tuning, really basic prompting to say, anonymize this, take out dates and addresses, and maybe replace them with something else, and then it works just as well as the current standard, industry standard NLP models. So again, projecting from there, imagine what that can do with a little bit better fine-tuning and prompting. So I think plenty of opportunity there. Moving on to a couple of interesting, what I think are interesting statistical methods and pipelines that have evolved in this space. Heterogeneity of treatment effects is an interesting one. It's been around for a while, but with machine learning specifically, we're seeing a lot more studies that have gone on to reinterpret otherwise negative randomized control studies. And the idea with heterogeneity of treatment effects is obviously the improvement or lack thereof is usually dependent on the individual's circumstances. And the idea here is, can I predict, either based on risk of disease or risk or prediction of a potential benefit in a patient based on the intervention, who's actually going to improve? So I'll pull an example out from critical care that's recently just kind of hot off the press here. This is a randomized control study. And so the randomized control study is comparing two methods of intubation, either bougie versus stylet. It's technical difference. One's a little more unique. The bougie is the more unique approach. And this is a randomized control study for about 1,000 patients, and the net result was negative. And so what these guys did is they took the RCT data, split it in half. For the first half, ran a causal forest algorithm to predict how individuals would respond to treatment, and then ran a validation study in the second half to demonstrate whether or not they've actually been able to predict if someone is going to improve with the intervention. And the figure on the top right-hand side gives you a sense for the overall negative signal at the outset, and then looking at these different groups of predicted treatment effects and how they vary over time. And you can see that in one instance, there's a treatment group that favors the bougie, and in the kind of further downstream, there's a clear favoring of stylet. And when then they look at these predictor variables that they used to develop this random forest model, they show which ones contribute to either signal, and it kind of makes sense when you look at it. It's, by the way, bougie experience plays a big role in intubation success, and patients that are potentially more hemodynamically stable or have a higher blood pressure, where you have a little bit more time, may also favor using a bougie. So again, just to give you a sense for the technique, now how do we take that and apply it to large cell observational data? And that's something that's obviously been done. I'd point you to the example from Legend Hypertension, which uses an OMOP-based observational database to look at the cardiovascular outcomes associated with either thiazide or thiazide-like diuretics versus ACE inhibitors. And then the spin on that study by Rekas et al., where they applied an HTE-based analysis, and where they did is they predicted the risk of a cardiovascular outcome like acute MI or hospitalization with heart failure, and looked at the effects within these individual strata to show that there was variation. So you can see that in the higher risk group 3, and these different colorings are basically just different databases that they used. There was a clear trend towards benefit in patients that were on thiazides versus ACE inhibitors. So this was obviously not a randomized control study, but really forced everyone to stop and question the heavy leaning on ACE inhibitors in the context of the management of hypertension. And specifically, sorry, these are for acute MI and hospitalization for these 2 love plots. And then the really cool thing, which is very classic for the Odyssey group, is then they took that and they put it into a pipeline, an extensible pipeline, and they shared it on GitHub, which is amazing, right? So standard data model, aggregate the data, reusable tools on top of the data. So I think this is a great example of where we're going. We can't talk about heterogeneity without thinking about place and heterogeneity. And this is work that we've done at Metro Health in Cleveland, looking at how much area deprivation contributes to tract level variation and death rates. And the answer is the majority, unfortunately. And what that points to is a substantial effect of racial segregation and socioeconomic deprivation on risk of mortality. And these are just some maps to show you kind of how it drowns out. The differences in the signal tend to drown out when you take out the area deprivation or the effects of racial segregation and socioeconomic deprivation. Where I think this all culminates, really, it's the most interesting, is this notion of a digital twin, which takes the concept of treatment heterogeneity and the richness of all the data that we have, both from electronic health records and the Internet of Medical Things, behavioral lifestyle, where you live, your neighborhood, and builds a framework and a model where essentially you have a digital twin, right? So when we ask the question, should you take pill A or pill B, we have enough data to model the probabilistic outcomes of either one based on a rich database at baseline. And so treatment A may reduce your risk of stroke, by 10%. Treatment B may reduce your stroke by 15%. And the converse is also true in terms of side effects. It's which one causes more nausea, for instance, in somebody like you. And so you can imagine, as we have deeper phenotyping from genetic material, that we get closer and closer to really understanding what a digital twin frankly looks like. A lot of talk about the space, but really haven't quite gone there for obvious reasons. It takes a lot of data and really advanced analytics and high quality data to make these calls. We are in Cleveland, so I'm thrilled to be a part of this recently approved R1 grant. We're looking at digital twin neighborhoods where the granularity is neighborhood, and understanding how public health interventions, for instance, we can model or simulate the impact of different interventions based on neighborhood with an eye towards health inequalities and disparities for specifically middle-aged individuals. And all of this, by the way, comes from electronic health record data. So I think some exciting developments there. As far as the last piece of this, I figured for platforms, I've gone through a couple. I don't want to beat them up too much, but having been exposed to a number of these, I can say what I like to see in a platform is really one that builds a scalable pipeline that extracts and harmonizes data with the least amount of effort required from the customer. And the customer here is an owner of a health database, or specifically in my case, electronic health record or health information technology ecosystem. I would love to see that data being really clearly versioned. That's something that we're not seeing a whole lot of. A lot of these networks just accrue data over time. And so every week you log in, it looks different. And there are probably really biased reasons as to why. So if a new customer comes on and their data is still loading, you can potentially make some bad calls. So along with clear versioning, clear data quality and provenance constructs should be a part of this. So excluding recent additions or excluding organizations that seem to have high rates of mismatch. So that needs to be a function there. I think the whole point of this, right, is bringing high quality data with up basically to the individuals that can do the most with it. So that's democratizing advanced analytics to end users and building technical solutions within the platform. And I'd say Trinetics is an interesting example of this, where they've built in the ability to do a propensity analysis on the cohorts right off the bat, so within their web platform. And that's been powerful. If you look at their number of publications over time, that's another one that's been impactful. Generative AI, if you guys have seen some of the work that, say, Microsoft has done in this space, you can go into Excel and ask it to run an assessment, and it can do the correct statistical test. So the risks of not knowing what you're doing aside, I think that that's a potential avenue to engage people with the data a little bit more. Thinking about how we handle the tension of privacy, both at the organizational and patient level. You know, I mentioned this with the Cosmos example. We've had to include or exclude variables based on an overall risk of re-identification. But if I'm trying to do a very specific analysis, it'd be really great if I could do that equation in real time, where if there are certain variables that I favor over others, that decision does not have to be made at a central level. So that's a bit more of an advanced functionality that I would love to see. From the organizational level, these platforms, you know, I would love to see more of a willingness to share data with the appropriate permissions, like public health entities. We would love to know more about their financial model. I mean, we know private, public, not for profit, but really, where's the money coming from? How much? Industry, otherwise. And then obviously transparent algorithms, quality checks and control. So, you know, an example there is for the Trinetics platform. They do a lot of natural language processing to bring in concepts and diagnosis codes, but those haven't been vetted, validated, or otherwise. And so I don't know how much you can hang your hat on those. Questions I haven't answered, nor do I necessarily have an answer for. I've far from ideal solutions today. Sorry. That's not me. Burning questions: privacy and permissions, who owns the data, who benefits, who's being incentivized by providing it, who's selling it, what is the threshold for de-identification with rich data? We know that's really dramatically different from just here are HIPAA identifiers. Are they in there? If you include enough identification data, you will find an individual. And then, you know, you need hours to talk about bias, fairness, and equity. And the basic idea here is these are not perfect observational scientific assessments or conclusions based on data. These are racist, you know, really broken views of a reality, which it's, you know, itself is compromised, and we can't keep that factory going. So we have to be really careful about what we do with this information and how we interpret the results and how we apply any interventions based on those results. And this is the grand summary, a little bit over 30 minutes of a whirlwind tour of my brain in the space. But the idea here from an operational research perspective is, I think it comes down to excellent data at the source, harmonizing and promoting interoperability, putting that all together onto a high value platform analytics tool, and actually democratizing access to that so we can get out of our own way and get smarter people to the data. And the general pressures that I talked about, so legislative, customer, AI, and novel statistical approaches are accelerating these processes. They're just ripe for all this disruption. But at the same time, got to be really careful. We don't have any answers, or no good answers, I should say, about privacy, data ownership, and bias. And so there's got to be a lot of work there. And I'll stop there. Thank you.
Thank you, Dr. Teravici, for a very interesting and amazing presentation. So we very much appreciate you bringing the informaticist perspective to this workshop. We have time for maybe one or two questions, and there is one in the Q&A. What criteria must be met to consider biological sample metadata as de-identified? You want to comment on that?
Oh, I don't have an answer. I already told you I don't have an answer.
Okay.
So it depends. I guess obviously it depends on the. Let me see if I can find that question. It's from Marianne, right?
It's actually from the audience, but I was actually going to invite. We still have a few members from our last panel on, and I thought they might also.
I would love that.
Have an answer for that. Okay. So if anyone wants to unmute to comment or just to answer it in the Q&A feature, that works.
So, I mean, I can say that the more context, it's right, the comment, the more context you add to a sample, And now we're talking about physical samples, which also potentially give downstream, right? So it's not just data you have today, it's data you could unlock later. The more metadata you add, the better it is from a secondary research perspective, but the higher the risk of re-identification becomes. And that's a continuous tension that we have to worry about. Another question, would LLMs also increase re-identification risk by drawing on wider data sources? Tell me more about that.
Well, I was just thinking that you showed very nicely how AI can help in especially name entity relationships. That was really cool, actually. But I was also wondering, and the de-identification, where it can very elegantly search through and remove items. However, we certainly run into this with the kidney precision medicine program, where we want to release more and more clinical side data, but with a small number of participants, we're limited for this. And I was just wondering if LLMs could be leveraged in
for evil,
a non-nice way to actually take a very small amount of information and start to re-identify people.
You're absolutely right. The answer is yes. The answer is they absolutely can be. And so this is, hate to say it, but it's basically the evolving war where you can do a lot of really good things, but then you can also do a lot of bad things. I think that we're kind of to some degree digging our head in the sand when it comes to this assumption that just by striking identifiers that are, the basic HIP identifiers, that you're not going to be able to re-identify. And I think the threshold should be much higher. And so my counter to that is, and I should have talked about this a little bit more about the digital twin concept, because I think what maybe isn't clear when you talk about digital twins, what you're producing is synthetic data. And I probably should have had a slide for this, but there's a lot of development in this space where I can take an existing electronic health record data source and other, a slew and variety of data points. And I can de-identify them by making them a synthetic data set. And that's more of a statistical concept where when you look at the data set, it looks like your data set. But when you zoom in on an individual person, it's not a person that exists in your data set. So synthetic data may help us. But again, I think that there's going to be a higher and higher threshold to prevent re-identification. And these tools are going to be able to, in more increasingly sophisticated fashion, find people. And that comes back to the whole privacy concept, which is, we're all data hungry and we want to keep smashing data together, but we really need to stop and think about engaging our patients and communities. We just don't do that enough to understand how they feel about this, right? You don't want to get into a Henrietta Lacks, parallel here, but that's that's essentially where things will go unabated if we don't pump the brakes and think about this more thoughtfully.
Dr. Terabici, unfortunately, we're at time, and I actually had quite a few questions for you, so maybe I will send those to you offline. But I do.
I'd be happy to. Yeah.
Go ahead. I do think, again, we're happy that you joined us today, because from the repository perspective, we are thinking more broadly about the relationships and the partnerships that we need to start establishing to actually more effectively build out the data ecosystem, right? And to leverage the resources that the repositories hold. So I know that our next session coming up is our workshop, where we're actually going to talk more about some of the new features that we're introducing to actually try to do that and to develop these partnerships and to have an impact on secondary research and this new generation of informaticists that are coming along and need to be our partners. So thank you again, and we'll look forward to continuing the conversation. And with that, we are going to move on to our second workshop for this workshop, entitled the NIDDK Central Repository Analytics Workspace Demo and the Launch of the Data Challenge Platform. And so for this, I'm going to welcome back Dr. Mike Culler as the moderator for the session. Dr. Culler is a principal at Booz Allen Hamilton with over 20 years of experience conceptualizing and implementing large-scale data sharing and analytics platforms. And he serves as the program director for the Booz Allen team supporting the NIDDK Central Repository. So Mike, handing it over to you.
Great. Thank you so much, Sharon. And I'm really excited to be following up on the keynote presentation that we just heard, to announce that Anya and Courtney, who I'll introduce in a second, are going to be demonstrating the new NIDDK analytics workbench. And not only talking about the analytics workbench, but it's going to be piloted as part of the first data challenge to be launched, reusing NIDDK CR resources. So I think it's very fitting that we're announcing this and launching the data challenge at the 20th anniversary workshop for the NIDDK CR, and also talk about some of the new functionality that we want to start piloting right away with the broader user base, such that, again, kick the tires, identify what works, what doesn't work, and ultimately move this analytics workbench into the production environment of the central repository. So turning to introducing our speakers today, pleased to introduce Anya Dabic. Anya is a health data scientist at Booz Allen Hamilton, and she specializes in scientific data management and stewardship. She has supported a number of different data management and sharing programs at NIH to implement the FAIR and trust principles for digital repositories. Some of these programs include the NIDDK CR, the NICHD Data and Specimen Hub, and the NIH ODSS RADX Data Hub. Her expertise includes health IT standards and technology, biomedical semantic standards, metadata models and schemas, data governance, and privacy-preserving record linkage. Anya has completed her BS in biomedical engineering at the University of Virginia. And then I'd also like to introduce Dr. Courtney Shelley. Courtney is also a health data scientist at Booz Allen, where she focuses on data science education and AI readiness of health-related data. She supported the NIH Office of Data Science Strategy to develop online data science learning resources for pre-college and collegiate audiences, and to assess data science education across U.S. universities to promote collaborative research between biomedical researchers and AI professionals. Prior to working at Booz Allen, Dr. Shelley worked at Los Alamos National Labs, where she received the Postdoctoral Distinguished Performance Award for COVID-19 response efforts at local, state, and federal levels, as well as conducted research in suicide prevention with the support of the VA and the Million Veteran Program. She completed her PhD in epidemiology with a focus on causal inference at UC Davis. So with that, Anya and Courtney, I will turn it over to you. Thank you.
Great, thank you, Mike. And I will go ahead and also share my screen for this portion of the presentation. Switch to presenter mode. All right, thank you, Mike, again for the introduction. Courtney and I are very excited to announce that the NIDDK Central Repository has launched its first data challenge centered around augmenting data for artificial intelligence. During this presentation, we will provide an overview of the data-centric challenge, as well as a demonstration of the new NIDDK workbench environment, which is being piloted for the data challenge, as Mike had mentioned, and will eventually be integrated as a permanent analytics resource for users of the NIDDK Central Repository. The aim of the data-centric challenge is to enhance data that has been shared through the Central Repository for AI-driven secondary research. The Central Repository plans to conduct a series of these data-centric challenges that are intended to build on one another to develop tools, approaches, models, and methods to increase data interoperability and usability for AI and machine learning applications. For this first data-centric challenge, NIDDK is seeking innovative approaches to enhance the utility of select NIDDK datasets that are centered around type 1 diabetes. The goals of the challenge are to generate an AI-ready dataset that can be used for future data challenges and to also produce methods that can be used to enhance the AI readiness of NIDDK data. This challenge will begin, is actually now open and will close on October 20th. So applications should be submitted by 5 p.m. on October 20th, in which participants will then have 9 weeks to develop their AI-ready data set for submission by the January 5th deadline. Researchers who are interested in participating can register through challenge.gov. As I mentioned, NIDDK is seeking innovative approaches to enhance the utility of select NIDDK data sets centered around type 1 diabetes. These data sets will be made accessible through the NIDDK workbench environment and includes data from the environmental determinants of diabetes in the young, or TEDDY study, which is a prospective cohort study that follows children with and without a family history of type 1 diabetes. The primary objectives of this study included identifying environmental factors that trigger or protect against the development of type 1 diabetes. Data from 4 TrialNet network studies will also be offered to participants. These include TrialNet-01, Pathway to Prevention, which is a screening and monitoring study that was established to provide a source of subjects for enrollment into the subsequent TrialNet studies, and includes children, participants ages 1 through 45. Participants from TrialNet-01 can then be followed longitudinally from a pre-disease state into an early disease state within the subsequent TrialNet studies. These include TrialNet-16, which is a prospective observational study that aims to observe the long-term effects of treatments used in the other TrialNet studies. TrialNet-19, which is a clinical trial that assesses 2 potential type 1 diabetes treatments, and TrialNet-20, which is another clinical trial that assesses the immune effects of oral insulin treatment. Individuals that are interested in participating in the challenge should visit the overview pages for these studies in the central repository to familiarize themselves with the study design as well as the data collected. The participation in this challenge will be tiered based on the challenge applicant's self-described experience with data science and analytics. The level of participation will dictate which data participants will be asked to enhance and make AI ready. So for the beginner level challenge, participants will aggregate data sets from the TEDDY study into a single unified and machine readable data set, and then enhance the aggregated data set for AI readiness. For the intermediate level challenge, participants will aggregate and harmonize the 4 studies within the TrialNet set of studies into a single unified and machine readable data set, and then enhance that aggregated and harmonized data set for AI readiness. And finally, for the advanced level data challenge, participants will first aggregate all files within TEDDY, and then harmonize the 4 studies within TrialNet, and finally fuse the aggregated TEDDY data set with the harmonized TrialNet data set and enhance that data set to produce for AI readiness. To judge submissions, NIDDK will ask data challenge participants to submit the following. The first being a single raw data set resulting from the data aggregation, harmonization, or fusion of all the study data files. The second being an AI-ready version of the data set that has been enhanced for AI readiness. Both the raw and the AI-ready data sets must be represented as a single rectangular file in CSV file format. Participants will also submit their code script that they use to generate the AI-ready data set, and a human-readable data dictionary that documents the variables in their AI-ready data set. And finally, they will complete the challenge solution submission form, where participants will document their AI-ready data set, the methods that they use to prepare their data set, and potential use cases for the prepared data set as it relates to type 1 diabetes or other disease areas of interest to NIDDK. I am now going to pass it to my colleague Courtney that is going to review some guidance for preparing an AI-ready data set, as well as the criteria that will be used for judging submissions.
Thanks so much, Anya. So I'm going to pop in and answer a couple of questions that you probably already have right now. The first one is AI readiness. So when we're talking about AI readiness, we're referring to data that is machine readable, that reliably reflects the study population from which the data was derived, are accurate in the sense that values are assessed against a reality check of plausibility, data that are explainable without referencing documentation that isn't included with the data set, predictive because the data accurately represents the conducted study, and finally are accessible for future AI applications. So to achieve these goals, you'll need to perform AI readiness steps, including typical pre-processing steps of addressing unexpected values, handling of missingness, relabeling and recoding of data elements and metadata for consistency, and especially to reflect standardized medical ontologies. AI readiness also requires extensive documentation, not only of data handling steps taken and descriptions of all variables, but also documentation of the data set itself, so future users can understand to whom findings can be applied. So when possible, best practices in AI readiness are to retain as much information as possible. If you need to transform continuous variables into categories for harmonization purposes, or you need to generate a new variable from existing variables, retain the original variables as well. So for example, when harmonizing two data sets where one contains height and weight, while the other contains only BMI, you can generate BMI in the first data set and then harmonize. This will necessarily generate some missingness, but will also retain information for future users. Real live humans will be assessing your submissions. We'll be looking for usefulness of your prepared submission materials. We'll look at your challenge solution submission form, which will include an informative description of your data set and the study or studies that generated the data, as well as descriptions of the data enhancements you performed and potential use cases for your AI-ready data set. We'll also be examining your code scripts and data dictionaries to ensure their usefulness for secondary applications. Pay special attention to documenting newly created variables, including the original variables used and the methodology used to create new variables, as well as the intended purposes for creating new variables, such as to allow for time to event analysis. I'm going to hand things back over to Anya for a little while now. Oh, wait, don't I have one more to do? Oh, I do. Okay, so sorry. I have one more slide. So how are we going to judge your efforts? First, we're going to assess that you submitted all the required components, including data sets, code, and documentation in the requested formats. Then we'll assess your submissions using a combination of human review and algorithmic review. We'll assess your data enhancement by algorithmically assessing metrics of accuracy, completeness, consistency, and validity. As previously described, we'll also examine the descriptiveness and informativeness of your human readable submissions. We will then assess the utility of your submissions for AI applications. Using your raw data set, we will pre-process using your AI-ready script to generate training and test sets, and apply a supervised machine learning model appropriate to your challenge level. We will assess performance using standard measures. And finally, we'll examine your potential use cases to see thoughtfulness and creativity. And now I'm going to hand it over to Anya.
Thank you, Courtney. So one winner will be selected from each participation tier, and each winner will be able to select from one of the following prize options. The first being to co-author a challenge results article with NIDDK repository and NIDDK data science staff to be submitted to a journal or a professional meeting. The next being to present the challenge results at an appropriate NIH level meeting, such as the Office of Data Science and Strategy, Data Sharing and Reuse Seminar Series, or a virtual meeting with NIDDK leadership to present the participants' challenge results. And all winners will be recognized as a challenge champion for the subsequent data challenges. The official rules of this challenge are posted on challenge.gov. By participating in the challenge, all participants agree to abide by these rules. Each participant must also agree to abide by the NIDDK Central Repository Data and Resources Use Agreement, or DUA, as well as the Data Challenge Addendum when registering to participate for this challenge. Each participant must also have a legal signatory authority from their institutions to enter into these agreements. If an applicant is not affiliated with an institution but wishes to participate in this challenge, they should contact the NIDDK support mailbox for further guidance. And finally, all analyses must be conducted within the NIDDK workbench environment, and participants are not permitted to download, copy, or extract data from this environment. The data challenge will be split into 2 phases. During the first phase, interested applicants will register to participate for the challenge via challenge.gov. In order to register, the following documents must be completed and submitted to challenge.gov. The first being the data challenge registration form, which outlines information about the participant, any team members, and their proposed research for enhancing the data for AI readiness. The second being the Data and Resources Use Agreement, which must be signed by the requester and the signing official from the requester's institution. And finally, the Data Challenge Addendum, which must also be signed by the requester and their signing official. Detailed instructions are provided under the How to Enter tab for phase 1 of challenge.gov. And as a reminder, applicants will have until 5 p.m. Eastern Time on October 20th to submit their applications. And we encourage applicants to submit in advance of this deadline to ensure that NIDDK has sufficient time to review and edits can be made before the registration period closes. During phase 2 of the challenge, which will open October 30th, participants approved to proceed to phase 2 will be notified through the NIDDK support mailbox with their AWS Service Workbench login credentials and detailed instructions on using the NIDDK Workbench environment. Data challenge participants will then perform data aggregation, augmentation, and enhancement tasks using Python or R programming language within a SageMaker Jupyter notebook to generate their code for making the data AI ready. So now for the demonstration portion of the NIDDK Workbench. So for background, the NIDDK Workbench is hosted by Amazon Web Services, and it is a secure environment that provides tools and computing resources for researchers to analyze the data. Within the Service Workbench, users will be able to launch Jupyter Notebook, which is a shareable document that allows researchers to write scripts to perform data cleaning, transformation, and machine learning using a variety of programming languages. And there is no cost to researchers to use the NIDDK Workbench. And so the goals of today's demonstration is to learn how to access the NIDDK Workbench, how to create a workspace within the Workbench, and how to launch a Jupyter Notebook instance within Service Workbench. I am going to stop sharing this screen and we'll go to the demo portion. So as I had mentioned, participants that are approved to proceed to phase 2 of the challenge will receive their credentials for logging into the NIDDK Workbench through their email. When they first click on the link to access the NIDDK Workbench, they will be prompted to log in and should use the password provided to their email when they were approved to participate in the challenge. Once logged into the NIDDK workbench, there on the left side pane, there will be two tabs that researchers will primarily use. The first tab is the Studies tab, which contains all the dataset files that researchers have access to, and researchers will only have access to the data files that are appropriate for their participation tier. And so under this organization tab, researchers will see all the studies that they have access to. Again, beginner participants will only see the TEDdy study package. Intermediate participants will only see the 4 TrialNet study packages. And advanced users will see both the TEDdy and the 4 TrialNet packages. The second tab is the Workspaces tab. And this is where users will deploy their own workspace to access and integrate the data. And workspaces are essentially collaborative computing environments where users can launch Jupyter Notebook instances and develop their code. And by default, users will also see this dashboard and SSH keys tab. These are more for administrative purposes and can be disregarded. And so to create a workspace and begin developing your code, you will first navigate to the Studies tab, and under the Organization tab, view all the study packages that you have access to. Then to create a workspace, you will select all the study packages and the data sets that will be contained in your workspace, and then click Next at the top of the page. Then you will select the computing environment, which will be SageMaker Jupyter Notebook, and click Next. On this page, users will provide basic information about their workspace, first being a name for their workspace. For demo purposes, I'm just going to call this TrialNet demo. Then under the project ID field, they will select their participation level, in this case, intermediate. By default, select the notebook configuration, and then provide a brief description of their workspace. Once all the information has been provided, we'll click on the Create Research Workspace button at the bottom right of the page. And then within the Workspaces tab, you will see that the workspace is currently being provisioned. And this takes around 7 to 10 minutes. And then this one should be launching soon. I'm going to do the demo portion of Jupyter Notebook using an existing instance. This one should be ready in just a couple minutes. I'm checking while we're waiting for this if there's any Q&As that have come up so far during the presentation. The first question is, does the applicant team have to be U.S. citizens or permanent residents? As outlined on the Rules tab in challenge.gov, yes, the challenge, at least one member from the team must be a U.S. citizen or permanent resident. And do any of the data sets include continuous glucose monitoring data? Yes, the TrialNet clinical trial studies include continuous glucose monitoring data, as well as the TEDdy studies. And to learn more about these studies, we encourage you to visit the links to the study overview pages in the central repository. And these links, these pages include overview information about the study, as well as the study documents, including the protocol code book, and other detailed information. And then, do applicants lose access to the data set they curated and the workbench after the challenge ends? So after the challenge, participants will be able to retain their code that they used to develop their solution in their private GitHub repository. The period of access to the data sets in the workbench is outlined in the data challenge addendum, and so participants will only be able to access the workbench and the data sets during the data challenge. However, if participants wish to continue working on their solution, they can create a separate request for the data after the challenge through the central repository and obtain access to the data that way. And by submitting a request through the central repository for the data, they will have access to the data up to 2 years. And Mike, did you want to, I saw you would like to answer a question live, or is that just by default in the Q&A?
Oh, I just marked it as that since you were answering it, Anya. Thank you.
Okay. So going to resume the demo. So here I have a workspace that I have already provisioned. And so to launch your workspace, you will click on this connections button. And under you will see the SageMaker notebook, and you will click on this connect button. And you will be navigated to this interface that provides a view of the file directory in your workspace, as well as active kernels or programs that you are currently running. From here, you will launch Jupyter Lab, which is the code editor for Jupyter Notebook. And so the Jupyter Lab, it's at the top right of the page, and you will click on Open Jupyter Lab. From here, you will select the programming languages that you would like to launch your Jupyter instance on. And the languages available include R, Python, and SparkMagick. So for the demo, I'm going to select R. And then you will see that a new notebook has been created. So on the left-hand pane, this folder icon is your Active Directory, which includes the data and documents that you have access to in the study package. And you will reference these file paths when developing your code. Under the circle icon, you can view all of the programs and kernels that you currently have actively running, and you can stop and restart your program from this tab or from the kernels tab on the top left pane. And then this third tab icon here, this is where researchers will be able to connect it to their GitHub repository and be able to asynchronously share their code if they are participating in the challenge as a team. And so researchers will develop and test their code in this environment. The Jupyter Lab interface allows you to write your scripts in cell blocks. And from the ribbon at the top, you can run, stop, and save your code. And so these options are available, or you can also run and save your code from the top ribbon under this Run tab. So this was just an overview of the NIDDK workbench, and a detailed tutorial will be provided to applicants that have been approved to proceed to phase 2. I am now going to turn it back over to Mike Keller to go over the Q&A portion of this presentation. Thank you.
Great. Thank you, Anya and Courtney. So again, if folks have questions, please do enter it into the Q&A panel. And as people are beginning to enter the questions there, I guess, I'll start with one question about, you mentioned data fusion earlier. What exactly does that mean? And what should I keep in mind when I go about fusing the data?
Sure. So let's start at the beginning of this process. So I first mentioned aggregation. So aggregating files refers to merging separate database files from a single study into a single tabular data set. So a single study was designed to be analyzed as a whole, it's going to be really easy to merge together files using some sort of column that all the files share, such as a patient ID column. For the studies we're looking at, the patient ID column is called mask ID. With the identified mask ID column, you should be able to merge or join your files using several methods in either R or Python on the workbench. Harmonization, it takes that up a step. So that usually refers to combining the same study over several years or several locations, such as combining census data over repeated years so that time trends can be analyzed. In this data challenge, we use the term harmonization to refer to merging several sub-studies within the umbrella of TrialNet. So since these studies are all very related, they include some of the same participants who were first identified in the TN01 study and then later appear in subsequent studies. We're looking at TN16, 19, and 20, when those patients were diagnosed with type 1 diabetes. So these sub-studies all harmonize well because they're reusing column names and patients. So when you're working in the intermediate level of our challenge and you're looking to harmonize these studies, you'll be looking for inconsistencies in column names and make adjustments there so that you don't accidentally create several columns that are highly correlated when merging. Finally, that leads us to data fusion, which is the process of integrating multiple data sources. So a classic example would be fusing medical data on children with asthma and other respiratory conditions with fine scale information on air pollution at the block level, where children live in order to understand each child's specific exposures. So neither data set would be able to answer such a question on its own, but fused together, we can add a new independent variable to a predictive model. So for this data challenge, we're aiming to fuse the TEDDY data set, which is a longitudinal observational study admitting newborns at high risk for developing type 1 diabetes, with the TrialNet set of studies, which admitted patients at a wider range of ages, but with similar inclusion criteria. So fusion of these two studies will allow for a wider study population, both geographically and by age. And the studies collected similar, but not always overlapping data. So column names will need to be harmonized when similar data was collected, but the metadata doesn't match by name. And this will occur more often when fusing than in an intermediate harmonization challenge, because while TEDDY and TrialNet have the same sponsors and many similarities, you're going to find many differences in naming conventions across the two large-scale studies.
Great, thank you. Question in the chat, and I'll bring that one up. What are the configurations of the environment in terms of CPU availability, GPU, and can that be modified based on requirements?
I can answer that, Mike, at least in terms of CPU availability. So each workspace instance will have a server size of 16 gigabytes, and that will be fixed. And so those requirements of CPU availability are set. In terms of GPU usage, I don't know the question to that now, but what we can do is post these answers to these FAQs to the challenge.gov page and send those out to participants after this workshop.
Great, thank you, Anya. And also just a quick follow-up, what about the programming languages that are available to analyze data in the workbench?
Yeah, and so in the Jupyter notebook configuration offers a variety of programming languages. These include R, Python, and SparkMagic. And so Python, including TensorFlow and SparkMagic, are ideal for more heavier computing, machine learning applications. And so these are available to participants to develop their code in Jupyter Notebook.
Great, thank you.
Mhm.
I guess, where I'd also like to turn to are if there are no further questions around, kind of the specification around the analytics workbench, want to turn to a little bit about some of the details around the review and the prizes that are being made available. Can you go into a little bit more detail about the algorithmic review criteria?
I sure can. So we derived data quality metrics from Erlanger and Voos, a 2022 publication, and a link is provided to that in the challenge.gov challenge overview page. So we're looking at simple metrics on different dimensions of data quality that are associated with less needing for data cleaning and pre-processing before use in AI applications. Specifically, we're looking at 4 metrics. The first one is accuracy. Accuracy assesses the number of errors in a data element that don't behave as we expect them to behave, such as an entry of 4 decimal point when we're expecting all numeric entries. That extra decimal point in there for a lot of languages is going to coerce that entire column to be a character column instead of a numeric, and then we won't be able to use it as numeric anymore. So someone would generally have to go in by hand and find that that missing or that errant entry and fix that before we could move on with using of this data set. Another metric we'll be looking at is completeness. So that assesses the number of records with incomplete data. This is an assessment of missingness, since many machine learning applications can't handle missing values in their input data. We'll be looking for consistency, which is a data quality measure that looks for consistent usage of language and labeling, especially spelling and capitalization. So an example of that would be a column titled sex of the participants. And if you're referring to that in terms of male and female, consistency would mean always using the same pattern of spelling and capitalizing, or not capitalizing letters as need be. So consistency basically means that you define your own rules for wording, and then you stick to those rules throughout your data set and through your codebook. The last data quality metric we're looking at is validity. Now that's closely related to the concept of consistency, except instead of you defining your own rules for spelling and terms, these spelling and terms come from a standardized ontology, such as MESH, medical subject headings. I wrote a lot more about this on the challenge.gov page, including examples. So head over to the challenge information page to learn more about these algorithmic assessment metrics.
Great, thank you, Courtney. And those links I see, in the chat as well, that we have been providing links to the challenge.gov website, as well as more information about the data, the studies, the data sets, and other news about the data challenge being launched. So please, I do encourage everyone to check out the links that are in the chat as well. So what about being eligible to win the prize? What needs to be submitted to be eligible to win a prize for the challenge?
Yeah, Mike, I can take that. So to be eligible to win a prize, participants will need to submit 5 items to be eligible for judging. The first, as I talked about, is the single raw data set that was generated from the data aggregation, harmonization, or fusion of the study files. This is again based on the participation tier, but this data set has not otherwise been altered for AI applications. And then using that data set, applicant or participants should also submit a AI-ready version of this data set, which has been enhanced for AI readiness. And both these raw and AI-ready versions of the data sets must be represented as a single rectangular file in CSV file format. And these data sets will reside in the NIDDK workbench within the workspace instance. And then participants will also need to submit their code scripts that they use to generate their raw and AI-ready files. And this code script will be submitted to their private GitHub repository. The next item is participants will need to produce a human readable data dictionary or codebook that documents their AI-ready data set. Excel format is preferred for this data dictionary, and at a minimum, it should include the variable name, the variable label or description, the variable type, the muted measurement unit, if that's applicable for numeric variables, and for categorical variables, the corresponding code lists and what the codes represent. And finally, participants will complete and submit to challenge.gov the challenge solution submission form. And this is where participants will document their AI-ready data set, what methods they used for preparing their AI-ready data set, and then describing what potential use cases they could use for their AI-ready data set as it relates to type 1 diabetes or other potential disease areas of interest to NIDDK.
Great, thank you, Anya. I have another question in the chat. Is a participant going to be working with one data set? And suppose TrialNet has multiple studies, so the proposal would be catered to a single study, is that correct? Or so is it one data set? Is it a single study? And what should the proposal look like?
Right, so a detailed description of each challenge participation tier is on the overview tab on the official challenge.gov page. So we encourage everybody to read each prompt in detail. But essentially with each of the participation tiers, the level of difficulty increases. And so for the beginner challenge for TEDdy, each of these study packages has their longitudinal studies and they have multiple data set files. And so for the beginner challenge with TEDdy, participants will need to aggregate all of the data files into a single merged or raw data set and then enhance that data set for AI readiness. For the intermediate TrialNet studies, participants are aggregating as well as harmonizing data across all 4 of those TrialNet studies. And participants in TrialNet 01, the same participant ID is used across these studies. And so that's how participants will be able to harmonize across the studies. And so for producing this single merged raw data set, participants will need to aggregate and harmonize across all 4 of the TrialNet studies. And then finally, for the advanced challenge, participants are aggregating and harmonizing to create a single fused data set that will then be enhanced for AI readiness. And so to participate and when developing your challenge solution and your proposed approach to developing the data set, you should follow the prompts for each of the participation tiers and merge the all data files associated with the studies within your participation tier. And participants, when you're registering for the challenge, you will self-indicate which participation tier you will participate in the challenge registration form, and that's also posted on the challenge.gov page.
Fantastic, thank you, Anya. Who is going to be reviewing participants' applications, and how will a participant be notified that the application was approved?
Yeah, so the NIDDK repository support staff and NIDDK program staff will review applications to participate in this challenge for completeness and relevancy to the challenge prompt of enhancing data for AI readiness. The NIDDK support staff may reach out during this application review process if any clarification is needed on the application. And participants that were approved to participate in phase 2 of the challenge, where they will receive access to the data in the workbench and and begin developing their AI-ready data set, they will be notified through the NIDDK. They will be provided at this time their credentials for the NIDDK workbench, a tutorial on using the workbench, and the tutorial and their credentials.
Yeah, great. Thank you so much. So, another thing, when I kicked off this demonstration, I had mentioned that the analytics workbench is a pilot environment right now that we will be launching with the data challenge. What happens with the workbench after the data challenge ends? Will I still be able to use it, or what's going to happen there?
Right. So as we had discussed earlier, and Mike, you talked about this, we are piloting the analytics workbench during the challenge, but the intent is for the workbench to be a permanent analytics resource for users of the NIDDK Central Repository. And so for future data challenges, we will be using this workbench and looking at ways to integrate it within the central repository so that our regular users will have the workbench available to them.
Yeah, and I think that's also an important, the other interesting comment you made is this is the first of a series of data challenges as well. So, this again, as we continue to mature the environment, as we continue to look at other studies, other data sets that might be available, other types of data challenges as well, this is going to be a continued rollout of data challenges in the future. So I would encourage folks who do participate in the first one, right, that it is worth keeping an eye out as new challenges are released in the future as well. So I do see we do have about 10 minutes left. Any other questions from attendees of the workshop today, or Anya, Courtney, any other comments to add? All right, with that, I see Sharon is back on camera. I will turn it back over to Sharon. Anya, Courtney, thank you again for presenting and demonstrating, and looking forward to the data challenge going live. Thank you both. Back to you, Sharon.
Yes, thank you, Mike, Anya, and Courtney. We're very excited about these new features for the repository, and starting with this first of a series of data challenges. So, but with that, we are finishing a few minutes early. So, Mark, maybe if we could put up the slide for the break. We do have a break until 12:50 today, and we can just begin that a few minutes early. But we'd like to remind everyone that they can stay online during the break, and we will resume at 12:50, at which time we're going to hear from investigators who have actually requested and successfully used DK-held data for new research. So please join us when we resume at 12:50 to hear from those investigators. So we do have people trickling back with us, but maybe we should get started in the interest of time. So thank you, everyone, for joining us again. We're actually resuming this afternoon with the second of our lightning talk sessions for this workshop, entitled NIDDK Central Repository Data Secondary Analyses, Qualitative and Mixed Method Studies. And so to chair this session, we have Dr. Ludmila Polakowski with us. Dr. Polakowski is a program director in the Division of Digestive Diseases and Nutrition at NIDDK, where she oversees a research portfolio on genetic and genomic studies of liver and gastrointestinal diseases, obesity, and nutrition. She also serves as the project scientist for the NIDDK Inflammatory Bowel Disease Genetics Consortium and as the NIDDK Extramural Genomics Program Administrator. Dr. Polakowski received her PhD in biomedical science and postdoctoral training in human genetics from the University of California, San Francisco. She was associate professor in the Institute for Human Genetics and Department of Anesthesia at UCSF before joining NIDDK. Dr. Polakowski is a member of the Repository Executive Committee. And Ludmila, I will turn it over to you to chair this session. Thank you.
Thank you, Sharon, and hello, everyone. I'm very pleased to moderate this session, and I will be fielding the Q&A afterwards. So please remember to put your questions in the Q&A box. So to begin, I am happy to introduce our 4 speakers in this session. The first speaker, Dr. Prasanna Santhanam, is an associate professor in clinical medicine and oncology at the Johns Hopkins University School of Medicine. Dr. Santhanam received his MBSS from BJ Medical College and his MD from the NHL Municipal Medical College in Ahmedabad, India. He completed internal medicine residency and a fellowship in endocrinology, diabetes, and metabolism at Joan C. Edwards School of Medicine at Marshall University. And he underwent additional training in nuclear medicine at the Department of Radiology and Radiological Sciences at Johns Hopkins to pursue his interest in nuclear medicine and molecular imaging of endocrine glands and disorder. His research focuses on new metabolic and molecular imaging techniques for endocrine tissues, artificial intelligence, and other predictive analytical tools for early risk stratification of metabolic disorders. He has used NIH Central Repository datasets like Look Ahead to investigate important research questions pertaining to cardiometabolic health. Our second speaker will be Ms. Juhi Chowdhury, who is the project and data manager of CKD-EPI-CT, which is the Chronic Kidney Disease Epidemiology Collaboration Clinical Trials. She is based at Tufts Medical Center in Boston. She supports a multidisciplinary global research group focused on using individual participant data to evaluate surrogate endpoints for future CKD trials. She has a wide range of experience in building and maintaining this large repository of data, from forming collaborations to harmonizing data for accurate meta-analyses. Ms. Chowdhury received pharmacy training from the Institute of Chemical Technology, India, and an MPH in epidemiology and biostatistics from Tufts Medical Center, Boston. With this blend of expertise, she is driven to study different aspects in the life cycle of treatments. Previously, she worked in the University of Baltimore-funded Needle Exchange program aimed at stemming the opioid epidemic in the Merrimack Valley of Massachusetts, and in a green technology project at the National Chemical Laboratory, India, to study methods for environment-friendly disintegration of runoff drugs. Our third speaker will be Dr. Sejal Mistry, who is an MD PhD candidate at the University of Utah School of Medicine MSTP program. She completed her PhD in biomedical informatics with a focus on machine learning, data mining, and translational informatics. Her research focuses on understanding the etiology of type 1 diabetes using advanced computational methods. Prior to joining the MSTP program at the University of Utah, Dr. Mistry completed her BA in biomathematics at Rutgers University and conducted pediatric malnutrition research as a Fulbright Scholar in Pune, India. And our final speaker, Dr. Joe Hedrick, is the Executive Director of the Type 1 Diabetes Consortium at the not-for-profit Critical Path Institute. A public-private partnership, the consortium is composed of industry and foundation members and academic partners and advisors from the NIH. Critical Path has aggregated type 1 diabetes clinical data from NIDDK, academic, and industry members, and works with global regulators to achieve broad regulatory objectives that will advance therapies for type 1 diabetes treatment and prevention. Dr. Kedrick earned his doctorate degree at the University of California, San Diego, and San Diego State University. Prior to joining CPAT, he was the venture leader for Janssen's type 1 diabetes program And he was also previously a senior director for cure and prevent therapies at JDRF and led a new drug discovery efforts at Merck and Schering-Plough. He is the author of more than 50 peer-reviewed research articles, reviews, and book chapters, and an inventor on over 20 patents. So now I am happy to hand the baton over to our first speaker of the session. Dr. Santhanam, please take it away.
Thank you so much for this opportunity. Hope you guys are able to hear me.
Yes.
So my talk is titled Variability in Adiposity and CV Outcomes in Type 2 Diabetes. Next slide, please. So overview of the talk: research, study hypothesis, methods, and results, scientific accomplishments, the key role that NIDDK Central Repository played in my accomplishments, my scientific career in the path forward, and then some helpful hints for mining these data sets and using these repositories. Next slide, please. So the research question was, is there an association between variability in adiposity markers and cardiovascular outcomes and deaths among adults with type 2 diabetes? What is the strength of this association? Does intensive lifestyle intervention influence the relationship between this variability and CV outcomes and mortality? So the study design and methods, we received the data from NIDDK repository after application and obtained the look-ahead trial data, the secondary analysis of a prospective cohort study of included participants in the action of health in diabetes. So the look-ahead was a multi-center randomized clinical trial that evaluated the effects of intensive lifestyle intervention compared with standard of care, that is diabetes support and education, on cardiovascular outcomes in persons with type 2 diabetes. The randomization groups were the intervention group and the standard of care group. The intervention group aimed to achieve a 7% weight loss or greater via increased physical activity and decreased calorie intake. The participants underwent a rigorous 4 to 5 years follow-up with very frequent measurements of their weight, and were coached on different physical activities and decreasing calorie intake and followed prospectively. A total of 5,145 participants aged 45 to 76 were enrolled between August 2001 to April 2004 across 16 locations in the United States. The trial was stopped in 2012. Look-at data was obtained from the NIDDK Central Repository, and we got Johns Hopkins IRB approval. Next slide, please. So the predictors were traditional risk factors and covariates, variability of BMI and waist circumference across 4 annual visits assessed using the coefficient of variation, variability independent of mean, and standard deviation. Variability independent of mean is a transformation of standard deviation, which is usually computed by the formula 100 times the standard deviation divided by the mean to the power of the regression coefficient, which is again calculated using the ratio between the natural log of standard deviation and the natural log of mean. The concept here is variability independent of mean is not related to the mean of the study population. The outcomes were all-cause mortality, cardiovascular deaths, deaths from MI or stroke, cardiovascular disease events, which is MI, stroke, and other causes. The covariates at baseline were used, which is age, sex, race, and ethnicity randomization arm, smoking status, alcohol consumption, duration of diabetes, and eGFR. The study group covariates were mean A1C, mean ratio of total to high-density lipoproteins, cholesterol, mean systolic and diastolic blood pressure, and use of blood pressure medications. Next slide, please. So the statistical analysis was stratified by the randomization groups. Characteristics were reported across quartiles of the coefficient of variation for each adiposity marker as mean or median for continuous or proportional for categorical variables. Cox proportional hazards regression models were used to calculate hazard ratios and associated 95% confidence intervals for the outcomes. 3 nested models were constructed for each variability marker, adjusting for different covariates. Model 1 adjusted for age, sex, race, and ethnicity. Model 2 was Model 1 plus adjustment for smoking, alcohol use, use of BP lowering medication, mean total to HDL cholesterol ratio, GFR, systolic blood pressure, mean A1C, and duration of diabetes. Model 3 was adjusted for variables in Model 2 plus further adjustment for mean BMI. If we are studying BMI variation, mean waist circumference in case of variability of waist circumference study, or mean weight in the case of weight variability. Next slide, please. So the results are outlined in these 2 broad tables. In the intensive lifestyle group, the people who wanted to achieve 7% weight loss with excessive diet regulations and physical activity tailored over 4-year period, the model did not show a difference in cardiovascular outcomes when we looked at variability in BMI. For example, in Model 1, Model 2, Model 3, the p-values were not significant, and the all-cause mortality did not show any difference in the intensive lifestyle intervention group when we were assessing for variability as a predictor, variability in BMI as a predictor for all-cause mortality. Next slide, please. However, in the control group, we see that all-cause mortality was significantly higher in all the 3 models, Model 1, Model 2, Model 3, when we accounted for variability in BMI as a factor, as a predictor for cardiovascular events and all-cause mortality. Cardiovascular death hazard ratios in Model 1 were greater than 12, in Model 2 were greater than 11, and Model 3 were greater than 15, all showing that variability in BMI is a very important independent predictor of cardiovascular mortality. Next slide, please. This study was also this study also showed that waist circumference also showed similar hazard ratios when you compared quartile 4 versus quartile 1. Variability in adiposity is associated with increased risk of morbidity and mortality in type 2 diabetes. Doing intensive lifestyle intervention mitigates that effect despite having variability in adiposity. So it means that if people pursue their goal of having 7% weight loss with diet and exercise measures, the variability in their BMI or waist circumference is not a factor in increasing cardiovascular mortality. Next slide, please. So scientific accomplishments, the NIH Central Repository is an invaluable source of high quality vetted data that is extremely hard to collect and obtain without extensive manpower and financial resources. The material provided to the researcher in terms of protocol, variable keys, main study, as well as ancillary study data is very comprehensive, has been very helpful for many scientific projects, and will continue to be useful in the future for myself. The staff are very responsive and prompt to queries about the application process, has helped me grow in my career. Next slide, please. As a first-generation immigrant, I would say that obtaining an NIH grant is tough unless you have a very good mentor with a track record of uninterrupted funding. NIH CDC repository datasets have been critical for establishing my credentials as a physician scientist. I've used the datasets to publish high-quality papers since these datasets have a proven track record of impeccable integrity and scientific rigor. Next slide, please. Some of the helpful hints are check all the necessary requirements for institutional IRB approval, ensure data safety, make sure your protocol to your respective institution addresses the issue of data safety and security, and especially cyber breach. Keep the NIDDK updated on the status of your manuscript, and strictly follow the acknowledgement guidelines that the NIDDK provides, so that all necessary parties are duly acknowledged for their contribution towards this data set. That's my presentation.
Thank you very much, Dr. Santanam. And a reminder to put questions in the Q&A, and all questions will be answered at the end of the session. So our second speaker is Ms. Yuhi Chowdhury. Please take it away. We cannot hear Ms. Chowdhury.
Okay, how about now?
Yes, we can hear you now.
Okay, sounds good. I can share my screen. All right, do you see the slides?
Yes.
Perfect, thank you. Well, so I'm calling the topic Revolutionizing Drug Development in Chronic Kidney Disease. So I hope to tell you how we've used the NIDDK data to advance how we bring therapies into the market for chronic kidney disease. So we're going to talk about the scientific problem, how we came up with the solution for it, what were the analytical methods, how the NIDDK data fits into this, our scientific accomplishments and lessons learned and conclusions. Sorry, I'm gonna take a quick break here. Are you able to hear me? I hear an echo, so I'm gonna try.
Yes, we can hear you. You sound good.
Okay, I will ignore my own echo. All right. So chronic kidney disease affects millions of people and disproportionately affects older population. And the end outcome, you must have heard it many times, is that kidney disease is a silent killer. You don't know until you're suddenly on dialysis. So, but it can be prevented. So you can see on this graph that, early intervention gives you more life years without disease. So, and yet there are still very few treatment options until very recently. And one of the reasons for having few treatments is ineffective endpoints in clinical trials. So an endpoint is basically something that tells you when to stop the trial and when you achieved enough efficacy of the drug. But in trials of CKD, they usually use like end-stage kidney disease, which takes many, many years to develop. And by that time, you need many participants, many resources for the trial to progress. And hence, that does not motivate clinical trials in CKD. But there is a solution, which is validated surrogate endpoints. Now, a surrogate endpoint is like a biomarker that can act as a substitute for a actual hard endpoint in a trial. And it occurs much earlier in the trial than a regular hard outcome, like, say, dialysis. And these can increase efficiency in drug development to slow the CKD progression. They can enhance feasibility of study patients earlier in the disease as opposed to later. So we're catering to a wider participant base. And this incentivizes product developers to increase their engagement to develop new treatments in kidney disease. So we're going to talk about how a biomarker becomes a endpoint. But before that, we'll talk about the few biomarkers that we use in CKD. The two most widely used ones are GFR and albuminuria. GFR essentially tells you how well your kidneys are filtering blood, and albuminuria is a measure of how much protein is being leaked in your blood, urine, which is not a good sign. And both of these are very useful tests that you can get from your healthcare provider GFR is something we should all know. So here's a plug for healthcare, caring for your kidneys. Know your GFR, ask your provider what your number is. And so this GFR is a indicator of kidney failure. And by that definition, it's on the path of progression to kidney failure. So you can see how markers related to GFR can be used as an endpoint in trials. So to study these biomarkers in 2003, so that's 2 decades ago now, we formed this scientific collaboration. I wasn't around then, but our lead researchers, Dr. Leslie Inker and Andrew Levy, received a grant from the NIDDK, actually. So we have this relationship for a long time with the NIDDK. And we founded this collaboration to study proteinuria as a potential surrogate endpoint in CKD, and as well as other epidemiological problems at that point. And over these years, we've had milestone scientific workshops. So in 2008, 2012, and more recently in 2018, with the FDA, with the EMA. So we're not keeping this as a siloed research aim. It's like, working with agencies, working with pharma companies, using the data to, generate more trials and more data. So we're actively engaged with all stakeholders. And so that through these workshops, we try to validate GFR and albuminuria as surrogate endpoints in kidney disease. And now I'll talk about the analytical methods and how the data comes into the picture. So there are 3 ways of validating a surrogate endpoint. First is, like I said, biological plausibility. And you'll remember how I talked about GFR coming on the pathway to kidney failure. So you need to have human studies to establish that. Then you need individual level association, which is epidemiological studies that tell you the association between these endpoints and the hard endpoint. And finally, we have trial level analysis, which is like the gold standard of validating a surrogate endpoint, is to show the treatment effect or like association between the surrogate endpoint and the clinical endpoint in actual trials. So that is where all these trials come into picture. And at CKD EpiCT, we include individual participant meta-analysis of these several trials. So there are multiple ways of showing your trial level association. You can, gather summary information from published papers and do association between the 2 outcomes that you want to test. But having individual patient data is so critical, is because you can have all the parameters that are missed out in a summary level statistic that you get from a publication. So you can get the errors in individual outcomes from the patient data. You can get like the confidence intervals are much more narrower if you have all these patient data. So what we do is within each study, we find out the treatment effect on the outcomes that we're interested in, so GFR slope and albuminuria, and the clinical endpoint. Then we, across these studies, we do a trial level meta-regression to see how the treatment effects on the surrogates relate to the treatment effect on the clinical endpoint. And we have a statistical base in University of Utah, and these very, very talented statisticians who help us, guide us, and also work closely with the FDA and EMA to kind of tell us what is the most latest approved statistical methods. So we conduct a Bayesian mixed effects meta-regression in the overall population, and then also study it by disease and GFR and albuminuria categories. And these models account for standard errors in the treatment effects and for the correlation on the errors in the estimated treatment effects between the different endpoints. So this really gives us a robust method of analyzing the surrogate endpoints, which we would not be able to do if we didn't have the individual patient data that we now have. And we right now have 66 clinical trials. And it's important to have trials across different categories of like subcategories of disease that you're trying to study in a meta-analysis, which gives you a high level of heterogeneity. And that is how you can apply it to a broad scope of the disease that you're studying. And if you remember from the 3 criteria that I was telling you about validity of a surrogate endpoint, one of them was epidemiological studies, but there the unit of analysis is individual people. But in the trial level analysis, the unit of analysis is individual trials. And in the disease area where there are already fewer clinical trials than we've had in other disease areas, it was such a challenge getting even these 66 trials. So we really, really want to thank the NIDDK and some of the other sources that we get data from to maintain these data sets, provide solutions to questions if we ask them. So from the NIDDK repository, we use the Hall PKD and the FSGS font data. So Hall PKD is a study of polycystic kidney disease, and the FSGS font is another type of kidney disease called the focal segmental glomerulosclerosis. And both of these diseases fit into that theory of having heterogeneity in the analysis and having trials in different settings. And even before the repository was set up, NIDDK-funded research or research data was shared with us by collaborators through their own goodwill. But the repository really brought a good structure and framework to how data are being shared, and that really helps the end user. So out of this, we published several papers, and we did like some simulation analysis, some trial level associations. But the key message here is how well the endpoint that we are studying relates to the clinical endpoint. So you'll see here the R-square, which is like a level of association between the two outcomes we're studying, or between the surrogate endpoint and the clinical endpoint, is 0.97, which is like 97% association between the surrogate and clinical endpoint, which is so one of the most highly supported surrogate endpoints in clinical trials right now. And all of this was possible because of, all these bubbles represent a study. So, the Hall PKD and FSGS font are a part of this collaboration or part of this data set, and they all contribute to this finding. And we don't just stop at a publication. We are taking it further, working with the regulatory bodies and trying to implement what we find in research to actual application. So we currently have approval from the FDA and the EMA for use of these surrogate endpoints in trials that are ongoing right now. So like Align, Protect, Applause, like several trials. And we also recently sent a request for qualification opinion to the EMA for use of GFR slope as a valid surrogate endpoint. And right now, I'm happy to tell you that the draft opinion has been approved and is open for public comment. They have this process where the public gets a chance to comment on the document, and other scientific bodies can also vet it. So it's now online. You can go and check it out. So now I'll talk about some of the challenges. And I actually had to go back to my code and find, try to remember any challenges that I faced with the HALT and FSGS data set, but I couldn't. They were very well documented. And we actually wrote a paper in JASN, the Journal of American Society of Nephrology. It is now accepted, and we talk about challenges in data sharing. But for the NIDDK data that we have so far, we were able to use the documentation, use the SAP, which is like the Statistical Analysis Protocol, and conduct research without much problems. But it is really important to have that documentation, because without that, we've had studies in the past where we had to, go around in circles trying to find out variables, or trying to see why variables don't give you the same results that are published. So all of this has happened with some of the other studies. So
we really appreciate having this platform. And it's also important.
Sorry to break it, but 1 minute warning, please.
Okay, yeah, that's perfect. And then we also suggest working closely with your grants and contracts team to keep these agreements always active. We, have a long-running program. We've been doing this for over 2 decades. So it's important to use the same study in the analysis as you go on. So, just constantly keeping your agreements active so that you can always use the same data set. And I think that is it. In conclusion, I'd like to point out how we use the data for validating these 2 surrogate endpoints, GFR slope and albuminuria, and how individual patient data really helps us solve these problems in CKD. And thank you to all the people who willingly share their data. So it's not just the participants who participate in these trials, but also the people who maintain the data and share it with us. And thank you for the FSGS and Hall team.
Thank you very much, Ms. Chaudhary. Our third speaker will be Dr. Sajal Mistry. Please go ahead.
All right. Thank you for that introduction. It's great. Okay. All right. Well, it's lovely to be here today to share with you all some of the work that I've done using the NIDDK Central Repository data. So my research focuses on understanding type 1 diabetes etiology by investigating factors that happen prior to disease onset. As most of you may know, the hallmark of pre-symptomatic type 1 diabetes is immune-mediated destruction of insulin-producing pancreatic beta cells. As beta cells are destroyed, autoantibodies can develop against proteins found within these cells, and there are 3 current big antibodies that are well-established biomarkers for pre-symptomatic type 1 diabetes, including GAD, glutamic acid decarboxylase, tyrosine phosphatase islet antigen 2, IA2A, and insulin or IAA. Current models to predict type 1 diabetes and subsequently enroll children in prevention studies consider these antibodies typically as binary markers. You have them or you don't. However, not all individuals that develop these antibodies actually go on to develop type 1 diabetes, and the time to disease onset varies considerably. There are several other antibody characteristics that may provide more precise estimates into risk and development of type 1 diabetes. Previous studies have established a relationship between risk for type 1 diabetes and the age and timing at which these antibodies appear, the type and combination of antibodies that appear in an individual in unison or together, and then also the titer or the actual measured value of the antibody at a given time point. And so taken together, while these studies provide insights into islet autoantibody development, few have actually considered the longitudinal effects of these characteristics here in unison. And so taken together, the focus of this work was to determine if a data-driven model incorporating islet antibody timing, type, and titer could stratify risk for type 1 diabetes in individuals with genetic susceptibility. And so to answer this question, we analyzed data from the environmental determinants of diabetes in the young, or TEDdy study, that was stored in the NIDDK Central Repository. This study enrolled 8,677 children with high-risk genotypes for type 1 diabetes across 6 different clinical centers. All of that data was collected and stored in the NIDDK repository, and this data was collected from birth until about 15 years of age or development of type 1 diabetes. And so what we did is we extracted data regarding the timing of different antibodies that were available and collected for all of these individuals, and we defined these 1-year cohorts marked as 1 year from birth all the way to the end time point of the study. For each of these participants, we extracted the types of antibodies that were measured using a standardized assay, as well as the specific titer of antibody and the measured value that were observed. So to analyze this data, we used a method called unsupervised machine learning, which is a type of machine learning. The goal of this method is to really infer mathematical relationships to identify patterns in data without making assumptions about the disease status. And so clustering is one type of unsupervised machine learning algorithm. This little depiction here kind of shows how clustering works. You have points that are measured in space based on different variables. And so clustering tries to identify subgroups in this heterogeneous data by looking at the distance between these different variables. And so we can see that these distinct clusters are formed. And then to wrap this all up, we performed a statistical analysis to evaluate risk for type 1 diabetes with the different clusters that we identified. And so overall in our work, we identified 6 distinct clusters of islet antibody development through our analysis that were present at various different ages. First, what I'll do is present what those clusters looked like, and then I'll discuss the ages that these clusters were identified in and their associated risk for type 1 diabetes. So presented here is the first cluster that we identified in this blue color. We named it the baseline cluster. To orient ourselves here, on the x-axis, we have the age. On the y-axis, we have the antibody levels, and this is a representative image here. And so each of these panels represents a different antibody. And so as we can kind of clearly appreciate here, this baseline cluster shows a low level of all 3 antibodies over the time period for this age group. Following that same schematic here, we have 2 additional clusters. In orange, we have an all declining cluster, labeled so because you can see that the levels of antibodies decline over time, which is most prominent in this GATA antibody. And then here we have the green cluster, which we've labeled all increasing, which again, we can appreciate that all 3 antibodies are increasing over time. The next 2 clusters were antibody-specific clusters. And so what we can appreciate here on this diagram on the left in red here is that this cluster has lower levels of GATA and IAA, but significantly elevated levels of IA2A. On the flip side, we see this purple cluster here, lower levels of IA2A and IAA, but significantly elevated levels of GATA. The final cluster that we identified here in brown shows lower levels of IAA, but higher, significantly elevated levels of GATA and IA2A. And so together, these findings highlight the 6 different patterns of antibody development with respect to timing, type, and titer that were identified in our analysis. And so what does this mean and how does this kind of develop over time? So depicted here are the different clusters that were identified in the different ages in our analysis. And so we can appreciate here in year 1 that we have 3 clusters of the baseline cluster, the all increasing and the all declining. In years 2 and 3, we only identified 2 clusters, a baseline cluster and an all increasing cluster. In years 4 to 8 of life, we identified 3 clusters, the blue baseline cluster, a purple GAD cluster, and this IA2A cluster. And then finally, in years 9 to 12, we identified 4 clusters, our classic baseline cluster, the IA2A dominant and the GAD dominant clusters, and then this brown cluster here that contained both high levels of GAD and IA2A. So the next natural question is, we found this delineation of patterns, but what does this mean for your risk of developing type 1 diabetes? And so presented here is a depiction of how the risk changes in each of these years with respect to a 1-year risk for type 1 diabetes. And so the big takeaways here is that before 3 years of life, risk for type 1 diabetes was primarily driven by belonging to a cluster where your antibodies were in this green or all increasing pattern. The baseline and the declining pattern had lower levels of 1-year risk for type 1 diabetes. The 5-year risk in this group approached about 50%. Then we can see a dichotomy happening here in years 4 to 8 of life, where we see that the highest risk is actually with the IA2A dominant cluster, and this GAD cluster has a moderate risk. Finally, in years 9 to 12, we again see a dichotomy here where we see the IA2A dominant cluster is really driving risk for type 1 diabetes, along with this GAD and IA2A dominant cluster, this combo cluster in brown. However, the baseline cluster remains a low risk for type 1 diabetes, and the GAD dominant cluster goes back to that level. And so taken together, there are several implications of this work. We identified these age-dependent risk profiles, which may be important for screening and enrollment and studies in the future. Also, this presents a very interesting question, which was the focus of my thesis research, which is why do we see these different patterns despite similar genetic risk? From a technical perspective, this work really shows how we can leverage machine learning methods to analyze this complex multivariate temporal data, but we also show how we can link machine learning with statistical methods for clinically relevant insights. In terms of the role of repository data in my scientific career, this access to this data and the NIDDK Central Repository has really made it possible for me to complete my PhD dissertation. From this work, I was able to complete the work that was presented here, but I also actually used the TEDdy study to investigate patterns of infection and sequences of infection over time with risk for type 1 diabetes. In terms of a broader perspective of my career, this research has really opened up really interesting questions on why these patterns happen, like I mentioned, and so we've generated some novel research directions, and it's also reaffirmed my interest in using advanced computational methods to understand type 1 diabetes etiology. And then finally, to end here with helpful hints for future repository users, my big takeaways are to really carefully understand data structures and the information that is collected and how it is presented. Also recognize that the repository offers quite a breadth of data available, not just specific to one pathology or one study, and also acknowledging the important role of the repository staff. They work very hard to ensure integrity of the data process and the handling process, but they are also very timely with their responses and actually facilitate communication with primary study coordinators when questions arise. And so I'd just like to end by thanking my mentors and acknowledging and thanking the TEDdy study as well as the NIDDK Central Repository for providing me access to this data, as well as our funding sources and the resources that allowed this research to happen. And thank you.
Thank you very much, Dr. Mistry. And our final speaker is Dr. Kedrick.
Yep, great. Are you guys able to see me?
Yes.
Okay, fantastic. Well, so first of all, really appreciate the invitation to speak here today. I think it's quite fair to say that the impact of the data that DK has gathered over the last 20 years really cannot be understated. And you'll see some of that here from our perspective and the work that Critical Path has done using in large part data that DK has aggregated over the years. So if we go to the next slide. Just a brief introduction to the Critical Path Institute. A lot of folks don't know who we are, but really the foundation of the Institute is our status as a unique neutral convener. We have a number of core competencies, data management and standards, biomarkers including qualification, modeling and analytics, regulatory and developmental science, and then clinical outcomes assessments. All of this feeds into developing drug development tools like clinical trial simulators, as well as advancing biomarker qualification. If we go to the next slide. I think this is a pretty typical data flow. A lot of what we do really is based on data, and our Data Collaboration Center is a critical asset here at the Critical Path Institute. This I don't think is a particular surprise to anybody. But this is sort of the standard data flow and workflow that we have with our data. And really the outcome are these actionable drug development solutions that undergo regulatory review at EMA or FDA, as well as other global regulators. The next slide. So I'm going to talk a little bit about some work, and I have a colleague who's listening here today, Dr. Kimberly Collins, who's responsible for quite a bit of this work, along with our academic collaborators. You should be seeing this published in Lancet Diabetes and Endocrinology quite soon. We don't yet have a date, but I'll only touch briefly on the data. The data that I'm going to talk about is aggregated from a number of clinical trials that were run under NIDDK and the TrialNet organization, but we've also been able to aggregate clinical studies from a number of industry and other academic sources as well. So this just gives you a little bit of an overview. So if we go to the next slide. So analyzing this data, in particular, we were interested in the relationship between C-peptide and HbA1c. This arose from prior conversations in a workshop with regulators with the idea of assessing C-peptide as a potential surrogate endpoint in type 1 diabetes. And again, this data will be coming out soon, and it was presented most recently at the Immunology of Diabetes Society meeting in Paris. But just a couple of key notes on HbA1c as well as some composite measures. HbA1c typically falls by 1% in the first 3 months, not surprising as these new onset patients go on insulin, and then inevitably rises in what we refer to as a J shape. Differences in HbA1c and intervention groups. So this is from in the clinical studies where active interventions are being used, are significant from 3 months on and persist out to 2 years. Differences in HbA1c are apparent even when HbA1c is low and improving. And we didn't see any particular advantage with any of the composites, the IDEX scores, beta 2 scores. The exception is that beta 2 doesn't require mixed meal tolerance tests, which is a relief of burden on patients. So that might be something to consider for clinical studies in future. If we go to the next slide. A key issue that regulators were concerned with is the proportionality of C-peptide preservation and HbA1c. This is shown here with a breakdown in quartiles with change in HbA1c represented on the y-axis and the percent C-peptide preservation on the x-axis. The top graph shows 0 to 6, 0 to 12, and 0 to 24 months proportionality. The bottom graphs show the same thing, except that we've left out the 0 to 3 months where HbA1c is pretty dramatically affected by the introduction of insulin. And you do see some interesting differences, but the conclusions really are important to draw that in the prospective studies, in positive studies where there was an effect of drug on C-peptide preservation, 24% difference in C-peptide was associated with a 0.55% difference in HbA1c. Whereas if we look at the cross-sectional studies, that proportionality was maintained, where a 27% difference in C-peptide preservation from baseline was associated with a 0.64% improvement in HbA1c. So the upshot is that C-peptide preservation here is indeed proportional with HbA1c improvements over all of the times that we looked at. And again, all of this will be presented in much more detail in this upcoming paper in Lancet D&E. So if we go to the next slide, please. So I did want to highlight the Type 1 Diabetes Consortium, which just as a note, Critical Path is funded by significant grants from the FDA. Much of the work of the T1D consortium has been funded by our consortium members, which include patient advocacy groups, JDRF and Helmsley, and a number of industry members over the years. We've been able to accomplish quite a bit, a lot of it enabled by NIDDK data. The collaboration launched in 2017 We have held workshops, most notably on C-peptide and its potential as a reasonably likely surrogate endpoint. Most recently, we achieved an EMA qualification opinion for the use of islet autoantibodies in prevention trial enrichment. So this is in trials in stage 1 and stage 2. And we have submission of briefing documents for a clinical trial simulation tool in new onset, as well as a letter of intent for a clinical trial simulation tool in prevention for stage 1 and stage 2. The data that we're using in prevention consists of 10 data sets, a large proportion of which are from the NIDDK data repository. We have a very large number of data sets from new onset. Again, a number of these are from the TrialNet efforts under DK, as well as industry and biopharm contributions, for which we're very grateful. In terms of modeling, we launched a publicly available clinical trial enrichment tool for prevention here recently in June. That's available. You can go to the Critical Path website to find those tools, as well as other tools that we've developed for other diseases. And I'll highlight a little bit of that in the next slide. And it was great to have Dr. Chowdhury presenting here earlier on kidney disease. I don't have to go into detail on that, but I did want to highlight some of the work of my colleagues at the Critical Path Institute in the Polycystic Kidney Disease Outcomes Consortium, where they've been looking at total kidney volume as a prognostic biomarker for PKD, as well as just recently, or relatively recently, achieved an FDA designation for total kidney volume as a reasonably likely surrogate endpoint for polycystic kidney disease. This was really driven by data from CRISP-1 and CRISP-2, as well as HALT. And that consortium is now working on developing a clinical trial simulator and a graphical user interface to optimize clinical trial design using total kidney volume as the endpoint. And if we go to the next slide, I think this is really a reiteration of what data has been used. The number of subjects, also looking at both eGFR and total kidney volume, and really highlighting here in red the data sets that we have acquired, Critical Path has been able to acquire from DK, without which this work really would have been impossible, both for type 1 and for kidney. So I think if we go to the last slide here, I'll just have a few comments. I didn't prepare a slide from lessons learned, but I do want to make a couple of comments. First of all, I'll echo all of my previous colleagues' presentations. I think there was excellent advice there. I think what we can add is the idea of planning ahead. Utilizing the data sets that are already existent can provide powerful insights into disease. You've seen a number of examples of that here today and throughout the symposium. And so the idea that going out and acquiring new data through clinical trial work, while I would never discourage that, I would encourage particularly young investigators that there's a wealth of data available to be utilized here without the expense and time of novel clinical studies. So I would certainly encourage you to consider that. I think the other piece of advice here I would give is you should be planning to do some work with the data. In terms of curating it, working with DK to understand, in particular with their data sets, understand what's there. It's not going to necessarily be ready for use immediately as it comes from DK, although they've done a tremendous amount of work in advance to minimize that effort. There's inevitably some work that you will need to do with your data modeling people. So I think with that, I'll just conclude and again, thank both NIDDK for their willingness to share the data that they've aggregated over the years, and also thanking all of those individuals who've helped fund critical paths effort, and in particular the patients that have all agreed to be part of these clinical studies, without whose participation we'd all be in trouble. So thank you very much.
Thank you very much, Dr. Hedrick, and thank you to all of our speakers for highlighting all the many different ways that repository data has successfully been used. A reminder, if we have questions for any of our speakers, please put them in the Q&A chat and we will ask them here. So maybe I'll start with a follow-up for Dr. Hedrick, but actually this applies to all speakers. So you are integrating data sets, multiple data sets, and harmonizing them. In what ways are you making that sort of harmonized and integrated QC data set, available for further secondary analyses?
Right. So most of that will be available through as we develop these graphical user interfaces. Because of the data sharing agreements, particularly with the industry and biopharma, we can't make the aggregate data set available, but people will be able to make queries of the data once those tools have undergone regulatory review. So an example of that is the clinical trial simulation tool for prevention studies that we made available with autoantibody data from DK and other sources as well. So that'll be the primary means for access.
And I can probably add, can you hear me?
Yes.
So the other way we are dealing with it, and I completely agree. So the agreements prevent us from sharing the aggregate data as well. But the way we are dealing with this is putting all of our programming codes on GitHub, so that once someone acquires the data set from a different source, they'll be able to replicate, the analysis pretty much very closely to how we have done it. So, right from the way we are cleaning the data set to our final analysis. So that's another workaround.
Also, there is a lot of information in the supplementary documents. For example, the JAMA paper, which we published, has a lot of information on the supplementary files and, the way the data was coded and everything.
Thank you. So going back to Dr. Santanam, how did you identify Look Ahead as the right data set for your study? Did you consider others, and are you considering replicating your results in another cohort? I think you're on mute.
Yeah. So I also am an investigator in the ACCORD study from BioLink. And first, our goal was to establish some initial findings from the look-ahead data, and then using machine learning, do cross-validation. Machine learning typically, involves, training and testing data sets with cross-validation, internal cross-validation, but then finally that will be applied to external validation for an ACCORD data set, which is again a large prospective study in persons with diabetes and looking at cardiovascular mortality and glycemic control. So that's one of the ideas, is to first, get preliminary findings, identify variables of interest, and then go on to do, AI generated, cross-validation and external validation.
Thank you. Again, a question that probably applies to several of you. So for those studies that combine data from multiple, I think up to 66 or more trials in some cases, to what extent are procedures for deduplication or identifying the same subjects popping up in different trials? To what extent is that part of the data, of your data flow?
I think it's potentially a little bit challenging with anonymized data to even determine whether or not there are duplicates. I think for Many of the studies, I think that's unlikely because of certainly in the new onset T1D, I should say. I don't want to speak out of school on the kidney studies, but with new onset T1D, you're sort of only in that new onset space once. And so, by the time you're done with most of these clinical trials, you wouldn't be eligible for another new onset study. So I don't think that's a particular problem for us. I think that's also likely true in the prevention studies, but that could be an issue. But since we received de-identified data, there's no way for us to be able to identify duplicate patients.
The one other
agree,
thing,
completely with that.
I'm sorry. Yeah, one other thing which bothers me is the repeated statistical analysis, involving a fixed data set where we run into the error of, multiple statistical testings and manuscript publications. For example, with a 6,000 people cohort, look ahead might be having 150 papers, and that's a problem. Yeah, when you are, even Bonferroni and these methods cannot account for multiple separate publications. There has to be some kind of way around these statistical issues where we will end up finding chance discoveries which are due to just repeated testing. I would like to hear other speakers' thoughts on this.
So the way at least we work with it is we keep expanding our systematic review every few years. So we include more studies and more diverse studies so that we are not always limiting our data set to this one set or like one pool of 66 studies. So we started off with 49, we are now at 66. We keep testing it in different scenarios, different disease settings or subsettings so that we know if our association is robust over time. So, the R square that I showed you on the graph earlier. So that's one way. And the other way is also working closely with your statisticians. And sometimes, there are things that as clinicians, we might not be aware of, but statisticians can really help you. So there's someone in our group working on missing correlations in some studies, like, what do you do? Or like, how much can you impute? Or like, what are the pros and cons of that? What are the pitfalls? So all of those things, how to make your models more robust. But I completely agree. Like you don't know with anonymized data if a person has been in multiple trials, but mostly because of the eligibility of these different studies, of the eligibility criteria, they probably aren't in multiple trials. But again, like as you mentioned, Dr. Santanam, the repeat testing, that's how we are dealing with it. Is like updating your data set, seeing if the metric is still robust in a different setting.
Maybe again, a question for everyone. So what did you feel was the most challenging aspect or perhaps the greatest barrier for new investigators in working with repository data? Getting back to advice for others who want to try this.
I can start on this topic. So I'd say for me, the biggest challenge initially was definitely getting a good grasp of the data structures and the presentation of what was available in the repository versus what I needed for the type of analysis or softwares that I use. And so my kind of advice for that would be to spend a ton of time looking at the data dictionaries and also reaching out to the staff should you have any questions or kind of clarifications on things. That would probably be my initial big place to start. I was very excited to hear, though, in the earlier session that there is a data challenge specifically for the TEDdy study available, because I would have appreciated that.
Thank you.
Yes, so one of the challenges is when you get a, say, CSV file or a a data formatted file, how to, aggregate the data and use SPSS, for example, or Python or R. So that is always the key, that when you are writing these codes for, say, machine learning in Python, and then you are removing the missing variables with one set of codes, then a lot of data can just disappear from the file. So these are some of the things which people have to really take care of and watch it very closely when they are using across different softwares or programming languages, where a data set will have a lot of missing values and it can disappear suddenly.
Yeah, I just have a note here from my colleague, Dr. Collins, who agreed wholeheartedly with what Dr. Mistry was saying, the data harmonization issues. And I think the other thing is also how you deal with missingness can be quite a challenge as well. So.
And the final question, what are you doing to make your results and your analysis available for sharing? How are you sharing your results?
Yeah, at least for critical path, I think, as I mentioned earlier, we publish everything that we do, as we can. We also obviously go to the regulators with the results of our analysis for approval, and then make those tools publicly available once they've received the regulatory imprimatur. So those are the primary ways through which critical path makes the results available.
The machine learning papers from Look Ahead are all posted in GitHub, as Dr. Chaudhary was mentioning. I mean, people are free. If they have query regarding the data sets, they obviously have to have prior permission and obtain the data set in the proper way. But I think posting the codes are a very important part of transparency. And
I think Dr. Hedrick and we are in the same sort of space, trying to validate endpoints. So we also publish our papers and we put them on a website that we have, and there's like a password protected section as well, which is a mechanism we've thought of for our collaborators. So like there are industry collaborators, so they can use some of the summary results, which are still not sharing individual participant level data because you don't have permissions for that. But before we publish a manuscript, there are these interim results which a pharma company can use just to even have some thinking or some discussion in their team, even if it's not vetted by a peer-reviewed journal. So it still keeps the science moving and keeps the drug development advancing. So that is another way we have kind of figured out. And then lastly, as you said, going to regulatory agencies, having the science not just remain in a journal, but actually help participants who need the treatments the most. Yeah.
I will echo everything that the other speakers have said, completely agree with all of that. And I think something that's rather obvious, but should still be stated, is an incredibly reproducible and thorough method section of any paper that we publish that's quite detailed in the steps that we take. And so I think that's the other kind of obvious thing here for reproducibility.
Thank you very much. So we are at time. So I'd like to once again thank all of our speakers for the stimulating presentations and for their work with the data sets. It's wonderful to hear about this. And I will hand it back to our MC.
Thank you, Ludmila. And I'd like to echo the thanks to the presenters for sharing their research and for encouraging other investigators to actually look to the NIDDK repository held resources for new research. And a special thanks to Dr. Mistry for giving a plug for our upcoming data-centric challenge. So please help us spread the word on that too. So with that, we're going to move on to our last session of the day and of the workshop, which is going to be a panel discussion entitled Supporting NIDDK-Funded Research and Expanding NIDDK's Data Ecosystem. And I'm happy to welcome back NIDDK Deputy Director, Dr. Greg Germino, to moderate this session for us. And Dr. Germino, we're very pleased to have you back for this final session. As you know, the session is actually structured to highlight some of the current activities with NIDDK projects and our partnerships with a forward-looking focus. So we're coming back to a lot of those things that you highlighted yesterday in your opening remarks whenever you went through some of the elements of the NIDDK strategic plan for research. So we're actually coming back to that now and are interested in how the central repository's role will actually help DK, achieve some of those objectives. So with that, Dr. Germino.
Thank you, Sharon. I just want to again congratulate you and the rest of your team on a terrific conference, really great workshop, some fantastic presentations. And it was great just to hear some of the examples of how people have really used our resources in meaningful and impactful ways, which is exactly why we funded this and support this in the first place. So that's really exciting. And I also wanted to put a plug in for that challenge competition. This is not only have we heard it could be helpful to people if we actually get this right, but I think importantly, also more broadly, is this a pilot for us to kind of help us think about what we should be doing in this, and again in the data space. And this is one way we can kind of test out how we can make this work better. And so really, the more people that play in that sandbox, the more information we'll gather and the more it can help inform our future. Because as Sharon just said, we are looking at our broader data ecosystem. And so this session is going to be focused on looking at that and how our repositories may fit with that. So we have three presenters and panelists who are going to first give us tell us a little bit about themselves and about their work. They'll each speak for about 10 minutes, and then we'll open it up with a general discussion and a Q&A section at the end. So I'm pleased to introduce our first speaker, Dr. Jeffrey Greth. Jeff, I've worked with a little bit through his work on DKNet over the years. He's the Associate Director for the Center for Research in Biological Systems at the University of California in San Diego. Dr. Greth has more than 2 decades of experience in providing collaborative data environments to biomedical researchers in order to advance scientific inquiry, leading to new discoveries and treatments of human disorders. He's the principal investigator for the NIDDK Information Network, DKNet, which serves the needs of basic and clinical investigators by providing seamless access to large pools of data, information, and resources relevant to the mission of the NIDDK. Dr. Greth has a BS from the University of California, Irvine in applied mathematics and a PhD in neuroscience with a focus on bioinformatics from the University of Southern California. Jeff, why don't you begin, and then after you have your 10 minutes, then we'll switch over to Dr. Noel Burt. So Jeff, please.
Thank you. So I'm going to give a brief overview of DKNet. So the NIDDK Information Network, next slide. As was mentioned, is really meant to be an information hub. So a single point of access to information about diverse research resources for research scientists, clinical investigators, including data, information on research resources, materials, tools, funding opportunities, various services to advance the mission of NIDDK. And so really, we're not providing, for example, computational infrastructure, but we're really trying to enable researchers to gain access to resources they might not be aware of, or to provide information on how best to use various resources. And a lot of what we've done, has focused on, providing, tools, services, information, support of rigor and reproducibility as well. So, one aspect is, we've been leading in the Research Resource Identifier Initiative, which provides identifiers for research resources, really to improve rigor and reproducibility of the research literature, and then also focusing on the fair data principles. And so this is takes new importance with the new NIH data sharing mandates, trying to help researchers increase the pools of data that are available for analysis by others. Next slide. And so think you have to hit. I don't think it was hit the. There you go. Okay. So again the fairness of an ecosystem. So being able to access information about studies that were completed doesn't just apply to data. It applies to, as I said, the resources themselves. And so this is a unique data set that we've been building around these resource identifiers, to help aggregation of information about key biomedical resources, which may, assist the formulation of new experiments, down the road, but also potentially, provide information about experiments data that haven't already been collected. And really, by being able to pull this aggregated data set together, we provide researchers with, unprecedented access to information, about the resources that have been used to generate data itself. Next slide. The other aspect regarding data is that, over the last, 5-10 years, there's been increasingly number of tools that have been made available to researchers, to do analysis online, to use online sources to investigate large data sets. And, that is sometimes a high barrier for researchers, for bench researchers, to gain access to these tools and to utilize them. And so what we've been trying to do as well is provide assistance in the set of, through the Hypothesis Center we have on DKNet, to provide some tutorials that we work with various projects, to bring information about these tools and about how to use these tools to access the data, to do analyses to the DK community. And, this is something, in the next phase of DKNet that I believe was also discussed yesterday, where, we're going to be increasing, our focus on, how best to provide, information access to these increasing number of computational tools, that can be used by bench researchers, to investigate, various hypotheses or generate hypotheses. So that's something else that we've been involved in. We've run a D-Challenge over the last couple of years, which has asked researchers not to perform an analysis, but to actually dig into data resources, such as data available from the central repository, to use this online data, online resources to generate new hypotheses that could then be tested with further experimentation. And so that's a challenge that we're also looking to bring back with this next phase of DKNet, to really, bring, researchers to, the data resources that are available, to use those, to further their hypotheses, their experimentation. And so, the Hypothesis Center, again, is one of these aspects of DKNet, that we're looking at building out. But really what we're trying to do is, again, DKNet is meant to connect researchers to the resources that are out there. So, connecting them to data, but then also potentially to, these online resources, where they can access the data, where they can do analysis on the data. Next slide, please. As I mentioned, increasing the pool of data that's available. And so DKNet, has been working, over the last couple of years, in building out our information around, fair data sharing. And so, we have an informational site on our portal, that focuses on the new data sharing mandate. We've also been running office hours, around this to help the community, start thinking about what this new mandate means for them, and, how best, to prepare for meeting these mandates. Next slide. Ooh, okay, so the areas that we've been focusing on, with DKNet, is really to increase impact, of research in DK and data that's being made available. So I mentioned, the Hypothesis Center. There are other areas of workforce development that we've been focusing on. So we've run a bioinformatics pilot program, trying to bring young investigators, and bioinformatics tools, into DK-related research. We have a summer program for graduate students, undergraduates, also focusing on data aspects, right? How best to manage data in the lab, how to utilize the data in the lab, because this is all key, in terms of building out these data ecosystems, right? That people have an understanding of what's required to ensure that as they start doing experiments, as they start collecting data, that they have good data management practices, so that that data easily flows into this larger data ecosystem that is being created, and that's really meant to help align all these NIDDK research efforts around fair practices. And so as we move forward into the next phase of DKNet, we're looking at how best to potentially also bring AI ML techniques into to bear against the data. And so, in closing, I mean, DKNet again is a unique resource in the sense that it is not, a core infrastructure builder, in terms of large computational data, hosting large data. But what we do is we bring people the resources to be able to access that data, where that data might be available, how best to use these tools, really focusing on the information about these resources, training and education around that, and then also really trying to push, the concepts of rigor and reproducibility, for resources and for data. So thank you very much.
Thank you, Jeff. So our next presenter is Ms. Noel Burt, who is the Director of Operations and Development of the Diabetes Research and Knowledge Portals in the Medical and Population Genetics Program and Metabolism Program at the Broad Institute. Her work centers on the operational and organizational leadership of large-scale international genetics consortia and public-private partnerships for human genetic studies, with a focus on type 2 diabetes and cardiometabolic diseases. Ms. Burt is one of the leaders of the T2D Knowledge Portal, or T2DKP, a consortium bridging government, industry, and nonprofit organizations. In 2020, AMP T2D expanded to include other complex diseases and is now known as the Accelerating Medicines Partnership for Common Metabolic Diseases, or AMP CMD. Ms. Burt trained in molecular biology and human genetics, and received her BS from the University of New Hampshire. Just a quick little plug for the AMP CMD. I have used it in my own work recently, looking at something in mice that actually it seems to have some human factors, human variants that would be associated with it. So it's really completely from a different field of science, and so it's really, really fascinating work. So anyway, Ms. Burt, please.
Excellent. That's a great introduction. So thank you for that. I'm really happy to be here. And I've got to say that having attended most of the sessions the last 2 days, I've learned a lot, more than I thought I knew about the NIDDK repositories. And this is really a fun experience. So thank you for having me. I'm going to pivot for this session. I'm going to talk about knowledge. And I think it's really fitting because I think we're using knowledge to try and strengthen NIDDK's data ecosystem. Our goal is to build a resource to have knowledge generated, to provide knowledge for all of these types of data repositories and resources that the NIDDK funds. So first, I want to talk about the cornerstone initial work that we did for the Type 2 Diabetes Knowledge Portal. And this has been around since 2015. And really, its main mission when we started was to make genetic association data available to the wider research community. Well, how do we do that? Well, we did it simply by thinking about the idea of presenting knowledge and results. What are we learning from all these studies for human genetics data for diabetes and its related traits? And we did this by really taking summary representations of the raw data, because the raw data is human, so it needs to be protected, and it needs to be stored somewhere safe and processed. And really the summary statistics, or the statistics that represent the association for the trait of interest, is really the knowledge that folks need to make decisions. And we did this in a framework that is circulated around scientific questions. So it is all question-based in terms of how we present this. And how did this all start? Well, we did this because we wanted to partner directly with researchers and the community and consortia and understand and do it exactly as a partnership. We understand how the data are transformed, analyzed, and represented. And then our goal is to radiate and make it accessible to others and share it with others so that others can use it. This really all started as we were funded by the NIDDK in 2010 to actually be a repository, the Data Coordinating Center for Type Diabetes Genetics Community. And at the time, we were generating large-scale sequencing and genotyping data. And we were very lucky to actually house the raw data. And we were building standards and practices for how do you process that data and represent summary representations. The entire community of complex trait genetics was doing this at the exact same time. So we're really galvanizing the opportunity to create standards. But we ourselves can use the data, but more importantly is you need to make this data accessible to folks who can create better treatments and better prevention models by understanding the biology. So for that, we created the Type Diabetes Knowledge Portal in 2015. And this really started in the sense that we partnered directly with these collaborators. We were actually working with them. We were part of them to represent the traits and the data standardized in the way that they intended to the broader world. And we started with 9 data sets and 25 traits. And what was really cool at the time is this was novel. No one was sharing summary statistics for human genetics data because you could re-identify them, people thought. But truly, we moved past that. We were very lucky, and the community started to take shape all along with us. Other communities like cerebrovascular disease, cardiovascular disease, type 1 diabetes worked with us and did the exact same thing, shared their summary statistics with us, and we produced them in a framework that is now called the Common Metabolic Disease Knowledge Portal, where this is an open access resource really for the knowledge of human genetics for common metabolic disease and its related traits, and for genomic data as well. And we really focus on 6 major disease areas. And the resource now, started with 9, now has representation of over 50 different countries, full genetic association data, fully integrated and accessible to you all, spanning over 500 data sets and over 500 phenotypes for common metabolic disease. So what are the sources and how does this all work, right? So the sources are threefold, and this is really important as the rest of how I talk about how we build knowledge bases. There's 3 things: partnerships. It's really important. The best thing is the human capital, the collaborators, the folks actually generating the data who understand the phenotype or the data type better than you do. And that's really the major source of most of our summary level statistics. Secondary, the world, curation and research. We listen to our collaborators, say what data sets should we bring into the knowledge portal. We do curation ourselves, we do automated research. And secondarily, partnering with disease agnostic resources like the GWAS catalog. We now have a resource built such that it's a 2-way sharing street with the GWAS catalog, who specializes in representation of GWAS data for all diseases, but we really work with them directly for common metabolic diseases, as well with the EBI in Europe to make sure data represented from all over the world is shared with the knowledge portal. So those are the 3 sources. And so the challenge is, though, that the field of complex trait genetics has done really well, right? So this is our chromosomes, and these are all the loci that have been associated with common disease. So I've named a couple that are specific to NIDDK's research. But what's important is we now need to pivot. We've discovered all these loci, but what we don't know is for these collection of variants in these loci, what the heck is going on there, right? All we know is there's an association with an organismal function, or in this case, a trait. There's this whole space in between, the regulatory elements, the cell states, the cellular functions, what is the gene? And this comes with a slew of questions, right? And so again, these are new scientific questions, but they have an increased complexity because in many cases with GWAS data, we were just dealing with really one data type. So how are we tackling that? Well, we need to integrate all this data and represent the knowledge. But each of these specific domains is its own research area, right? Instead of investigators have their own sort of way of doing things, way of representing data, and in many cases, repositories or resources. So I've just mentioned some of the types of data we need there, but the approach that we're taking is exactly the same, right? We want to summarize the knowledge coming from these different data exploration activities, framing them by representing summary representations and encircling that in scientific questions to represent the knowledge. So how we're doing this, our approach is very simple. We collaborate directly, as I mentioned before. We understand the data. We first start with what is the knowledge? What is the scientific question? And then we work back to what is the data needed to integrate? What are the analytical results needed? And then we onboard any pre-computed results very easily, make it very low bar, very free and fast for contributors to use, and pull summary representations into the knowledge portal. We can also encode any method that is out there that we can use to generate summary representations of raw data, and we incorporate external resources. If we do all this successfully, we want to make this integrated in a way that's really easy, a workflow that steps you through the different types of representation of the results from each of the data that is framed in scientific questions that we hope is intuitive. So now I'm just going to step you through a couple examples by which we take knowledge and really frame it in a scientific question in a presentation, but really allow you as a researcher to unwind all the way back to the primary data. We do this across many of the NIDDK resources. So this is the gene FTO, many people know if you're studying common metabolic disease. And this right here summarizes all the genetic evidence we have from all those data sets that I mentioned before, the GWAS studies and the sequencing studies, all integrated and represented in one table, which summarizes all the common and rare variations. So it's all the phenotypes, all the genome-wide associations and the gene level associations for the single gene. So this summarizes the genetics evidence. But in terms of time to integrate and ask other questions, if you scroll down the page, what if you want to know this tissue-specific gene expression for FTO? What's great about this is these are results. But what we've done here is also integrate both disease-agnostic resources. So you have Blueprint, GTEx, and ENCODE integrated here for their summary and gene expression levels. But also you have disease-specific resources. In this case, HPAP is an example. So this is an NIDDK-funded resource. Not only do we pull together the data, but then if you actually want to drill down into the actual results, you can scroll through the results, find the actual summary information, all the provenance of that data, then directly go to another NIDDK resource, which represents these results and these data even better because they focus on specifically the donor and the data that was generated. So this is all the primary data. So this is just one example of how we're starting to allow a user to see the knowledge framed in a scientific question, and then allow you to wind back, because in many cases, there's so many layers of data integration that go into one question, right? So we're doing this in other aspects. You heard earlier, we were talking about biodata catalysts. There's another resource on the portal where you can actually run a customized analysis on summary representation of data that's sitting in biodata catalysts. So the individual level data is there. We generate summary representations and you can compute them through custom filters on the knowledge portal. Another resource that's coming online, integrated fully in the portal, is the KPMP, which you heard about earlier. So a way forward for us as a resource, and I think this is part of the conversation we'll have later, is really resources can get really antiquated really fast, and maintenance isn't really useful. Really, maintenance equals sustainability. You've got to keep it relevant. A resource has to be constantly pivoting to the scientific questions that the field is asking in its space. So to do that, some things. You need to capitalize on the efforts of data repositories like NIDDKs, all these disease-specific and domain-specific resources. We need to connect with them to understand what the data they have and work together with them to create summary representations. We need to create crosstalk across these resources. And I mean this both in human and machine-based space. Human in the sense that we need to talk to folks in these resources so we understand what they're trying to do, but also create ways for the data to flow both ways, the results and the data to be connected. Create structures for sharing within these resources. Instead of a pull-based where you have to ask everybody for data, make it push-based, to do this through APIs and standard callings and standard data paradigms so you can share data readily. Allow resources to be computationally accessible. We've heard about this earlier today. I love the data challenge because this really encourages user-defined analyses. You need to make it easier for those users to contribute back to the resource so that knowledge can be shared, because it's really the knowledge that most non-domain specific experts are trying to use. So a person in industry does not know to have the raw data from human genetic studies. They want the summaries, right? Create new models for contribution, build ambassadors. What I mean by that is that anyone who contributes, in my mind, is a member of the portal. So they get to go out and talk about it, but they're also part of our community. And that's really exciting because that builds incentives for sharing. Orchestrate the community to develop scientific questions. Really, this is really important as we enter these more complex data sets and questions, because it's going to require multiple levels of data integration. And therefore, that amplifies all the other underlying resources and makes all resources better. I think it's important to embrace generative AI tools, but we must insist on human expert curation. So this is really where you leverage back the community. We run these very elegant AI tools, but also make those results accessible so that they be honed and perfected by the experts. Finally, sharing and educating. Really, I think as not a data scientist by training, frankly, I think the most important thing is human capital and working with the investigators directly to understand what they want in a resource, what knowledge is important to them, and then we can go out and figure out how to represent it to them. So with that, I'll stop sharing and turn it over to the next speaker. Thank you very much.
Thank you, Noel. Next speaker is Dr. Rebecca Lee. Dr. Lee is the CEO of Vivli, and on faculty at the Center for Bioethics at the Harvard Medical School. Previous to her current role, she was the executive director of the MRCT Center of Brigham and Women's Hospital and Harvard for over 5 years, and she remains a senior advisor at the center. She has over 25 years of experience spanning the entire drug development process, with experience in biotech, pharma, and CRO environments. She completed a fellowship in 2013 in the Division of Medical Ethics at Harvard Medical School. She earned her PhD in chemical and biomolecular engineering from Johns Hopkins University. And Vivli is one of the 7 generalist repositories participating in the NIH Generalist Repository Ecosystem Initiative, or GREI. Dr. Lee, please.
Thank you. I'm delighted to be here and talk about Vivli and our partnership with NIDDK. Next slide, please. So today I'll be introducing you to our platform or repository Vivli, how we've implemented the FAIR principles for sharing of clinical trial data, and what we're doing with NIDDK to expand the ecosystem. Next slide, please. So the Vivli platform, and if you can just click through twice. The Vivli platform is, we're a nonprofit entity. We offer convening for both the industry, academic partners, nonprofit funders and foundations, as well as the government. So primarily NIH and others, as well as patient and patient advocates. Our primary mission is really to lower the barriers to share clinical trial data. We oversee the implementation of a platform that is also called Vivli. It's a user-friendly, secure, state-of-the-art data sharing platform built on Microsoft's Azure. We serve the international community, and we include trials from any disease, country, sponsor, funder, or investigator. But we are starting to collect a number of communities, trials from many communities that include collections. For example, we have a number of trials in the area of diabetes, COVID, and oncology, and these are becoming larger and larger collections in certain disease communities. Next slide, please. What I'll talk next is just spend a minute on different approaches to sharing of human data. So there is 3 main approaches. So open access, where anyone can access the data, and there might be simple account creation and sometimes a data use agreement. The second type is managed access, which is the type that Vivli uses, or a gatekeeping process. So data in Vivli is kept in a gatekeeping process where data is accessed for scientific purposes only. There is an independent review process, and some of the data can be only accessed within a secure research environment or secure sandbox, and there's a clear legal framework within which the data can be accessed. Other types of data is in what we call restricted access and invitation only. Next slide, please. So when we think about these 3 different frameworks from accessing data, there are other types of platforms as well for trial data. So we have broad and generalist repositories that accept all types of data regardless of therapeutic area or domain. We fall in kind of this broad or generalist repository. So we are one of the generalist repositories that is supported by the NIH. Some others are listed here. And there are some more specific or disease-focused ones. We also have another repository in this category, which is focused on antimicrobial resistance. Next slide, please. So the next thing I wanted to focus on is who contributes data to Vivli. So we have 2 categories, and the reason why this is important is that all of this data can be integrated together. I'll give you a few examples. So we have a number of industry members. So about 2/3 of our members are members of the pharma industry. So if you think about all this data, and we think about big data, so I know NIDDK is, for example, interested in type 1 and type 2 diabetes. So for example, if we look at GSK, they are also contributing quite a bit of data in that area. So if you look at the next slide, we also have members of academia, platform and foundation members, and this is where NIDDK's data comes in, as well as, for example, the Helmsley Charitable Trust. So they have also contributed several large type 1 diabetes trials that they have also sponsored, as well as others, for example, from Harvard, UCSF, Hopkins, and other foundations that do not fall in the pharmaceutical area. So we can think about any of these data sets on Vivli can now be combined together. And this is the only place as an example, if you're interested, for example, in type 1 diabetes, you can combine data from NIDDK, Helmsley Charitable Trust, and GSK, just as an example, if that scientifically makes sense to you. So you could do that within our platform. You can make a request, combine that data together, and sign a data use agreement and combine that data together. So that's what people are doing every day in different areas, oncology, COVID, and of course, diabetes. Next slide, please. So I'll talk a little bit about the FAIR implementation by us. We have 3 search modes to enable scientifically precise queries. Trial data is described by standardized metadata and terms from the Cochrane Library, and data from multiple data contributors and platforms are made accessible through a single process and able to be combined. So my kids have just gone through the college application process, so I can just describe that as the Common App for anyone who knows what that means. So it's one application and you can access data from any one of these contributors. It's supported by a secure research environment and pre-populated by common analytical tools. And that's how this really works through us. So you can put in one application. Next slide, please. So what is included in the data package made available by researchers? You would receive the study protocol, the data dictionary that describes all of the variables, the SAP, something called this clinical study report that's given by the pharmaceutical companies that really tells you all about the efficacy and safety data, the IPD, which is actually the individual participant level data. That's really the raw data that underlies the clinical study, and then the anonymization report that outlines how everyone anonymized the data. So you have an idea of what you're getting, and then some optional pieces as well, such as the case report forms that could be extremely useful. So that's what's included in everything that you'd be getting when you access data from Vivli. Next slide, please. So findability, this is a very clear search. We come in, put in some keywords. You can do some refining of sponsor type. For example, you just can type in NIDDK, type 2, phase 2, or GSK, or just simply search on the sponsor type, start dates, or by phase. And you can come up with what you're interested in and just put in a request. Next slide, please. So NIDDK and Vivli, how are we partnering? So 93 studies are listed on Vivli that redirect back to the NIDD platform. We also have NIDDK studies directly archived on Vivli. The requester is redirected to NID for processing and approval, and the governance and data use agreement remains intact. So that's how that's really working. We have integration with over 7,000 other data sets on Vivli, and so that is what we think of as big data. So that data can be integrated directly with other data that's on Vivli. Next slide, please. So thanks so much for your attention. I'm happy to answer any questions if there are any.
Thank you, Dr. Lee. So we can maybe have all 3 panelists. We can just turn off the slides. I don't want to read the slides anymore. And so now we're going to open it up to questions for any of you that are out there in the group that have been watching. We've heard 3 different ways that the different panelists, their platforms are intersecting with NIDDK activities. You know, we heard one where providing kind of integration of resource discovery and information discovery that's relevant to NIDDK and to bioscience. Heard one where various genetic studies and phenotypic studies are being linked together and clustered in a way that you can do novel discoveries. And then lastly, where you can use, again, a place to go look for clinical studies and participant level information from not only across NIDDK's clinical repositories or data repository, but even the broader universe. So lots of different ways that you can think about the data science space. So just a couple of, while we're waiting for questions to come in, we had a couple, one here that just came from Ravi. Is Vivli providing support to investigators, institutions, in creating their data management and sharing plans through payment?
So we are providing support, and we do get so many questions about the DMSP. So we have templates available on our website. I'm not sure what you mean through payment, but we do have those templates for payments and how that works. We don't charge. I mean, so if that's the question, we do have a budget template that is available on our website.
So Robbie, could you clarify in your question what you mean by through payment?
Yeah, I mean through contracting. I mean, if the PIs pay money for you and then you do some service for them with the drafting DMS and all the way up to submitting their applications.
Okay, so if you're asking if it costs money to archive data on Vivly, yes, there's a single payment that's available if you want to share data on Vivly. It's fairly modest. It's several $1,000s for a single payment. For members who are part of us, like Harvard and UCSF, that's all in one single. If they're members, they don't pay anything. And hopefully that answers the question. But I think that's all in the budget template. So if you download the budget template, it should all be within there.
Thank you.
Thank you.
So a question for you, Jeff. So you've heard, you've mostly kind of collated information from from our data sets around a lot of particularly our basic science data. How do you see yourself intersecting with our repository, our central repository, either now or in looking to the future?
So I think, I mean, there are two ways, I think, primarily, that we're intersecting. I mean, right now, actually, through our office hours around the new data sharing mandates, the DK repository is one of the places, where, data should be deposited, right? And so there is this, providing investigators with, the appropriate information about, how to meet their data sharing mandate requirements, under the new, the new regulations. Moving forward, as in the next phase of DKNet, we are going to be bringing on more sort of computational type tools, to bring to the community and also build out sort of the, as I mentioned, the Hypothesis Center, the educational component about how people access data, how people access, bioinformatics tools. And I think that's another area where tying, further into the central repository, and other repositories to, allow bench researchers the ability to get to these resources more easily. I think is critical moving forward. I mean, bioinformaticists, are very important in research, but, I think there's more that, we can provide, like what Noelle was showing in terms of, what the knowledge portal does, really makes this data, more accessible, to bench researchers who are willing, to explore, potential hypotheses they have or, genes of interest, and sort of building out that part of the ecosystem, really making these data repositories, bioinformatics tools accessible to the broader community.
Thank you. So this is a question for actually all three of you, because you've discussed in your ways. I mean, obviously the value of data is the more you have and the more different types you have, the more information, interesting patterns may be discovered. And so to build the various data sets or the knowledge bases that you have, you've had to get people to partner with you. Establishing relationships, partnerships, collaborative, or even competitive approaches. So if you could comment on what you see are the kind of the key elements being able to successfully do what you've done, including having people even have data like in Europe, right, where they have really tight sharing restrictions. I mean, so how have you kind of negotiated this space?
I can start. I think it's a coalition of the willing. So I think we've been very lucky. I think, well, currently the best thing is publication because of the new challenges. Because if you want to publish the results, I think that if you make it really easy for them to share their results and help them publish and do that in rapid time frame, that's been really helpful to have like people contribute. So that's for contributing. But in terms of partnerships, that's actually been really fun, particularly because 5 years ago, or actually 7 now, when we started the knowledge portal, there weren't actually a lot of knowledge bases, so we were unusual. And so therefore, as other as resources are new, they're still getting their legs established and still trying to establish themselves as a brand and a resource. So sometimes it's actually kind of challenging to partner with them because they have a mission. But what I've noticed over time is actually people are learning that, actually it sounds cheesy, but we're better together, right? So if there's something you're an expert in and I'm not, you can actually say, well, let's create an API so that my results come to you. APIs and our ability to build standards make it easy. So you need to make it easy. You need to give them some sort of incentive so that, there's some sort of, both parties get something beneficial out of the relationship. And you also need to be able to sort of respect each other's domain. I think that's important. So for us, like a lot of times we're talking about other people's data. We're stewards. And I love that theme of like NIDDK data stewards. We're knowledge stewards and we can't get it wrong. So we're very careful. And so I think if you're careful and you try and do things also simply, you're going to get more people interested in working with you. So that's been kind of our approach, slow and steady. But it's been quite fun, frankly, learning from the community.
Rebecca, Jeff, either of you have anything to add to that?
Yeah, I think something that resonated with me about what Noel said is certainly to understand what everyone else's mission is. I think it is difficult to get people to share their data, and that's what we're trying to do. We particularly have to work across different stakeholders, pharma, academia, and foundations, and to get each one of them to really hand over their data has been a bit tricky over the last 5 years, but to make sure that what we're doing is developing a trust relationship and make sure that we don't break that trust and work within their mission to make sure that they get what they need out of this relationship and that their incentives are aligned with their mission. Sometimes, the collaboration doesn't always work, and sometimes it takes several years. So being patient before we work through certain collaborations.
I mean, that principle of making sure that they can see there's some reward, right? That's the broader challenge we're having, even as we think about implementing that new NIH data sharing policy, right? What are the rewards for people to share? It's not always, as you just said, people don't, it's hard to share, and sometimes it can be hard because you have to have all the proper, metadata and everything else captured properly. There's lots of reasons people don't want to share, and so incentivizing them is really obviously an important part of that. And so I'm interested in how you were able to do that in pharma, for instance, which historically have been relatively reticent to share individual level participant data.
Yeah, so it's very interesting, and I'll tell you in a nutshell, the trade associations, pharma and FPA, came out with a position policy, which is called the Pharma FPA principles. And so since they all aligned with those principles, they basically, anyone who's part of those trade associations have to share. And so they basically don't have much of a choice, and they feel like they've committed to transparency. Those principles are all about transparency. Mm-hmm. So they've kind of committed to transparency. So they have a gap in and they have to share their data transparently. And this might be a mechanism for them to share through our platform, or they may have to do it on their own.
Got it. Thank you. Mm-hmm. Jeff, and about your experience?
Yeah, so I mean, I think, as was previously mentioned, I mean, I think, critical thing is trust. And also, what are the benefits, for any of the parties involved in the collaboration? For many of the things we do, we're also sort of brokering information between different sources. So also, providing, sort of the role as an honest broker between, potential repositories that people are going to work with, is also very important. So that, we can, make sure that, what a group is trying to do, they can do that successfully and are, plugged into the right resources. So again, it's communication, building trust, and then just, making sure that, everyone understands, that, everyone sort of has a stake in the game, and, everyone is seen, as sort of someone who should have, some, skin in the outcome, right? So that we're all working well together.
So one of the, I think it was, I'm not sure if it was Noel or Rebecca had mentioned sort of sustainability, right? So, you have to keep it, you have to keep it current. You can't just build it once, right? This is an ongoing process. And so related to sustainability, these things are not inexpensive. And we certainly have talked about this in a broader scale with other data sharing policy implementation, for instance. So how do you think about that in terms of establishing the right, partnership with, whether is it government, industry, whatever, as you think look to the future, particularly if interests, go elsewhere?
I think there's a couple models we've thought about. One is, really trying to minimize how much data you're storing, because that's really data storage and compute is a massive burden, right? So for us, we're not a data repository. So the data repositories have the harder work. Although when you're dealing with the genome, you have lots of variants and we are getting a bigger footprint. So for us, we try and, do things cleverly on the cloud with compute, but we try and leverage other things, right? Other existing technologies, other existing resources. But I think in terms of, we want to make the resource always open access. That's like super critical to us. I never want someone to pay for access to knowledge that someone else generate. I think that's not a good way to do this. So what we're considering is like options for like, sort of like you contribute, you get, if you want to build like a workspace for running analysis for people, where you can actually allow them to do compute on the data and then make the results accessible. If they contribute the data back, it's free, for example. But if they want to make it private, maybe they have to pay a month. So that's like, if the hose were shut off from NIDDK and we had to sustain the resource, that might be a clever way to do it. There's other ways to do it where you do actually partner with foundations, people who have very specific interest in a disease area. But really, it's about, the best thing we've learned is about pivoting to different types of scientific questions, because we're very lucky in our field that we're always going to have more scientific questions, and you can really build off of that if you have a really key framework. So that's really how we approach it.
Yeah, I mean, one thing I would want to second from what Noel mentioned, and as I mentioned, we've been doing a lot of sort of office hours around the new data sharing mandates, and the question of big data always comes up, right? So if labs, are collecting, this large scale data, I think that is going to be an issue in terms of, down the road, how does that get sustained? Because again, the larger the data gets, the larger the cost. So, really working out, what of the large data, needs to be maintained. So maybe not all the raw data. So thinking back, looking at sort of, the physics community, which does, similar things, right? They're collecting a load of data, but then they do sort of initial triage of that data, of what gets stored and shared down the road. And I think that's something, moving forward, as data sizes get bigger, as, images, get more resolution, that's going to be critical to try and figure out, in terms of sustainability.
Thanks. We had a couple of questions come in from our audience. One is just following up on the data policy. And so the question is, for those of us at NIH who have to manage the DMS policy, are there key needs you're hearing about that aren't being met by existing resources?
I can speak to that, but it may be fairly focal to clinical trial data. So our data is, when it comes into our platform, is anonymized, required to be anonymized. And one need is that NIH does not, at least to my knowledge, reimburse for data to be anonymized. And our other foundation partners do have that as an allowable cost. So we feel like the barriers for those other foundations that allow that to be an allowable cost has been really lowering the barriers for data to be shared. But for NIH, that is not apparently an allowable cost.
Thank you.
Mm-hmm.
Any, Jeff?
Oh, I was just going to say, I mean, I think, one of the critical area is, as also data sets containing, multiple modalities, are being generated, where does that data go, right? So it's not a question about, is there an existing resource potentially? I mean, we always have the generalist repositories, but, with NIH's policy in terms of trying to also make people use the community repositories, which have, specialized tools and, ensure metadata compliance for, the various data types, how do researchers navigate that? So I think it's, maybe not a question about individual resources, but, helping researchers start navigating the whole ecosystem of data repositories that are out there.
I'd say that's one thing, sort of knowing what's out there, what options they have for existing resources. I consider myself knowledgeable in the resources, and I learned, about NIDDK resources in the last 2 days, ones I didn't know existed. So that definitely speaks to the fact that either I haven't been paying attention, which is possible, but there's a lot out there, and some of them may fit people better than others. So I think making helping guide the researcher to which existing resource they might want to deposit in or work with may be a little bit useful to help people with, because we've gotten some questions about that, because we're also been helping people with the data management policy, because a lot of the results are actually really important, and we have really nice structures to make those accessible beyond the repositories that are actually better for the raw data. And we've fielded a lot of those types of questions, so that would be the one place I could imagine being very helpful.
Thanks.
And also, actually, one more thing, feedback. Actually, this is actually a question, sort of, as I've been writing data management policies myself, data sharing plans, feedback from the NIH are how are we doing at writing them? Are we responsive? I know that it's super new, so, but sort of what's the word, pitfalls, or things that people get wrong when they're writing their data management and sharing policy, sort of having that sort of, to populate, maybe an FAQ page or something would be really helpful too. Maybe people overthink it maybe a lot of times, and that might be, that sounds kind of simple, but that could be a helpful thing to do too.
Thank you. One last quick question, and this was actually specifically to Rebecca. It was related to one of the points on your talk, I guess, and that is, can you clarify your last point of DK datasets integration with other 7K datasets? Apparently that was somebody had seen that come through, and I didn't see that during the talk.
I'm sorry, you said NIDDK integration with
your last point of, DK datasets integration with other 7K datasets, I guess, in your collection of data that you have.
Yeah, so if you're asking if NIDDK datasets can be integrated with our other datasets that are on Vivly, the answer is they can be. Yes.
Okay. Great. Mm-hmm. Thank you. We are at the end of the time. I want to again thank our panelists for their really great discussion, great presentations, really giving us again the full spectrum of ways that again in the DK data science landscape can be leveraged and can be effective and impactful. And I'll again turn it over to Sharon to close us out. Thank you.
Thank you, Dr. Gemino, and I'll echo your thanks to the panelists for a very good presentation and discussion. So a great way to end the two-day workshop. So now I think Rebecca Rodriguez, our repository director, is going to rejoin for a few closing remarks.
Yes, good afternoon, everyone. And thank you all for joining us these 2 past days. And thank you all the speakers and the panelists for getting this accepting to speak. We had a great 2 days. And right now I'm going to attempt to summarize briefly the outcomes of today. And this morning we discussed the applications of data and biospecimen standards. We went and discussed quickly throughout the application of data, both for data and biospecimens throughout the entire life cycle. And we discussed that throughout the entire continuum of fairization and to have a continuous data augmentation of resources. We also then heard an interesting talk about innovative approaches for secondary use of data resources, including some EHR, from an informatist perspective that would take advantage of existing resources and technology for data standardization, aggregation, and analytics. And this will achieve interoperability with evolving platforms. And then after that, we had our launch, our first in a series of data challenges. And what we hope to accomplish with that is for enhance our data for future AI applications and assets. This will hopefully be the first in a series. And this will, for us, build on one another with the potential to increase the pipeline of secondary research. And we also demoed our pilot workspace, what we are calling our alpha version. And we are asking that if you are participating, please give us any feedback from the data challenge in the platform, and this will help us make it a lot better. Then we later heard from some of our requesters and the great work they have been able to accomplish with the resources they have requested. And all the value added, not only to the resources that we hold, but also to the impact that they have done to their career progress. Finally, we just now heard from a panel of experts that discuss different platforms that support NIDDK-funded investigators, and also various ways that these strengthen and expands our ecosystem, as well as great potential that we see for them to establish new partnerships and relationships to build out and in parallel develop tools and technologies that influence the new generation of informaticians. And with also outreach and training opportunities. And it's clear that there's plenty for us to do now. Next slide, please. I want to remind all of you that the slides and the recordings will be available on our website. And you can visit our website many different ways. We're putting a QR code that you can snap with your cell phone right now. You can also visit, we're putting the direct link here. You can also reach out our inbox. And if you had direct administrative inquiries, you can reach us out, and our address is here as well. Next slide. Again, a plug-in, I cannot say it enough, for our data challenge, and the QR code for the direct site at challenge.gov is here as well. And stay tuned, because we will have updates for these and the data challenge series throughout. Thank you so much again, all of you that attended this two-day workshop, and all the speakers and panelists for graciously accepting the invite and for your contributions. It's been a great 2 days. Thank you.